Good day, everyone. I want to welcome all of you to the 4th International Cohorts Summit of the International 100K Plus Cohorts Consortium, or IHCC. My name is Jeff Ginsberg from Duke University in the United States, and I am co-chair of the IHCC along with Peter Goodhand of the Global Alliance for Genomics and Health. In terms of logistics for this meeting, you've all gotten emails with instructions from our team about using this virtual platform, so hopefully that's all clear. This is a really exciting time for the IHCC. We're 3 years old, and like toddlers of that age, we are eager, energetic, and exploring all kinds of things. In 2018, we had our first summit at Duke University. We had 100 attendees from about 60 cohorts, 25 countries, and representation of over 30 million participants from around the globe. At our second meeting, we had a meeting in Reykjavik where we committed to engaging cohorts from low and resource settings and low and middle income countries. In 2018, we had 4 cohorts from these regions using World Bank definitions of low resource settings and low and middle income countries. And today, I'm very pleased to report that we have 28 cohorts from low resource settings and low and middle income countries in the IHCC. In fact, today we are nearly 110 cohorts from over 45 countries and nearing 60 million participants worldwide. So we've nearly doubled in size over the last 3 years. Later today, you'll be hearing more on how the IHCC is working towards more engagement of inclusion of cohorts from South America, Asia, and Africa. And we will be hearing from cohort leaders in these regions on how they hope to work with the IHCC. I would like to highlight a few of our accomplishments in the past year since we last met and recognize our stellar secretariat. Eric Plummer, Hannah Cannell, Ludi Dementon, Meredith Towery, Chris Nelson, and Teji Rocker-Burris have all kept us focused and very productive as we could be during this global pandemic. We completed and ratified the IHCC Charter and Governance Model, and you can find these on our website at ihccglobal.org. I want to thank Mary De Silva from the Wellcome Trust and John Connolly from CHOP for spearheading that effort. We also finalized our policies for data sharing and industry engagement. Those two are on our website's resource center. And I want to thank Laura Rodriguez and her team for making that happen. We launched the IHCC Global Cohorts Atlas, a key strategy to make cohorts and their discoverable, as well as to help us find their metadata. And I want to thank Thomas Keane, Melanie Court, Christina Young, and Philip Awadala for leading this effort from the European Bioinformatics Institute and from the Ontario Institute for Cancer Research. We solicited last fall research proposals for cross-cohort science and were able to fund 6 of them, covering mental health, opioid use, obesity, and the COVID-19 pandemic. And I'd like to thank Hakan Hakanarsson, Adam Butterworth, and Katrina Federico for developing a peer review and selection process, and also for enabling the engagement of nearly 30 cohorts in these projects worldwide. We are also working with the Davos Alzheimer's Collaborative to create new approaches to diagnosis and treatment for that disease. And again, thanks to Hakan for his leadership there. And we completed a 5-year strategic plan and have a new vision for the IHCC, which is creating a global community of cohorts working together to advance science and improve health for all. And our tagline is linking cohorts, understanding biology, and improving health. And you'll hear more about this tomorrow. In this meeting on day 1, in a moment, you'll hear about the recent project progress in more detail on the data architecture, the scientific strategy, and the policy strategies that the IHCC is undertaking. Tomorrow, we will highlight the strategic plan for the next 5 years and engage all of you in helping craft the specifics of what the IHCC should do going forward. And on Thursday, we'll focus on engagement as well as the role the IHCC might play in global pandemics. We also have 4 amazing keynote speakers. On Thursday, we will hear from Francis Collins, the director of the NIH, and Ambrose Wonkum from the University of Cape Town. Tomorrow, our keynote speaker will be Mary De Silva from the Wellcome Trust. And today, it is my great pleasure to introduce our opening keynote speaker, Dr. and Professor Corey Bargmann, president of Chan Zuckerberg Science and an internationally recognized neurobiologist and geneticist. Dr. Bargmann is also head of the Lulu and Anthony Wang Laboratory of Neural Circuits and Behavior and the Torsten Weissel Professor at the Rockefeller University in New York. She is a member of the National Academy of Sciences and the American Philosophical Society. In 2012, she received the Kavli Prize in Neuroscience and the 2013, the Breakthrough Prize in Life Sciences, among many of her scientific honors. She has also co-chaired the National Institutes of Health Committee that set the goals for the strategy for President Obama's Brain Initiative. She's a former Howard Hughes Medical Institute investigator, and she has a PhD in biology from the Massachusetts Institute of Technology. Dr. Bargmann is the Chan Zuckerberg Initiative have been incredible supporters of the IHCC, and we're really grateful for that support. I want to welcome Dr. Bargmann to this meeting to open our sessions on. She'll talk on accelerating science through technology and collaboration. Dr. Bargmann?
Good morning. Good morning, Jeff. Good morning, all of you, and thank you for that very generous introduction. It's an honor to be here today. It's an honor to be included with the IHCC in their international meeting. I came to the meeting in Duke in 2018. I was impressed with what they had done then, and I've just been considerably impressed throughout the passing time with what you've accomplished and how you are bringing people together and the sophistication with which you're doing that. I'm going to talk today, next slide, about the work that we're doing at the Chan Zuckerberg Initiative. Jeff asked me to talk a little bit about what we have learned from the pandemic through our work. And I think that's a really good framework for thinking both about what we're trying to do at this new philanthropy, which is just a year or so older than the IHCC, and also how the IHCC and the CZI think in common about common problems. So the mission of our group, which was set by Mark Zuckerberg and Priscilla Chan, our founders, when they decided 5 years ago to start this philanthropy, was that they wanted to support science and technology that will make it possible to cure, prevent, or manage all diseases by the end of the century. Now, that's a very bold goal, but in thinking about it, it does actually make some statements about what their priorities are. First of all, it's science and technology to really work on the foundations that make it possible to make progress. Second, it's all diseases. Don't try to think about things one at a time. Try to think about things systemically and make progress in many directions as broadly as possible. And of course, that is exactly what this kind of cohort work is doing now at a direct-to-patient and medical level. So in terms of thinking about what we could do, we decided that the best thing we could possibly do to meet a goal like this was to accelerate biomedical science, to make every scientist more effective, to allow every scientist to make progress faster. And from that perspective, we think the two most important things that we can do are to develop new tools and technologies that accelerate research and to support open and collaborative models of research that allow people to work together to make progress toward new goals. Our values are people, technology, collaboration, and open science. If I could have the next slide, please. We try to make an effort to accelerate biomedicine through a variety of different mechanisms. We fund grants and requests for applications in areas that we would like to pursue. We're currently funding scientists in 30 countries through this. We also have one aspect that's a little different from most other philanthropies, which is that we have an internal technology group with software engineers and product and user research people who come from the Silicon Valley culture. It's led by Phil Smoot, the head of science technology and the vice president of engineering at CZI. And this is actually helping to build different kinds of tools that enable our work on science, taking some of the advances that have happened in the commercial sector, but have not yet made it back into the nonprofit domain. We collaborate with other funders and with other groups, and we also engage directly with society through a movement for science and through working with patient groups. Next slide, please. So what we think we're doing right now is a set of experiments in accelerating science. We're building tools and resources for and with scientists, and we're thinking about ways of changing the culture of science to make it more collaborative and to recognize more players in the scientific enterprise. I've listed our starting programs here on the right-hand side of the slide, and what I'm going to do is talk about a few of those programs, specifically what's happened in the past year, in the COVID year, and what we have learned from that about how to accelerate science that we didn't know before. So next slide, let's start with a project that started at the same time as the Chan Zuckerberg Initiative, and that is the Chan Zuckerberg BioHub. That is a technology-focused research site that brings together scientists, engineers, and physicians. It links together 80 faculty members from UCSF, Berkeley, and Stanford University, the three great Bay Area schools. And it also has in-house groups that are working on technology development and on infectious disease. Its co-presidents, with the spirit of collaboration between schools and between disciplines, are Steve Quake, who comes from bioengineering and applied physics at Stanford, and Joe Geraci, who comes from infectious disease, the Department of Biochemistry at UCSF. Now, this institute was working on a variety of problems in March of 2020, when we realized that there was a nationwide emergency and inability to test for the presence of the new virus in people who are coming down with respiratory diseases. Now, this is a technology site. We have a lot of platforms and robotics and barcode printers. And in addition, we have an interest in infectious disease. And so Joe Geraci, together with UCSF, decided to set up testing capacity within the BioHub. The first thing we learned from this is the importance of partnerships. If I could have the next slide, please. Partnerships, relationships, and collaboration were absolutely critical for doing what made the BioHub special. So first of all, at the BioHub, we had a number of highly trained professional people and equipment who were able to set up testing very quickly. The Chan Zuckerberg Tech Group that I mentioned to you, led by Phil Smoot, was able to build the kinds of software systems that could then interact and process and follow the samples of 2,000 people per day. So that was very rapidly spun up. And that created capacity. But very soon, we realized that the important capacity in this area was not necessarily serving the Bay Area hospitals, but rather serving underrepresented people in the Bay Area community. And we learned that through our partnership with UCSF, who helped us set up a CLIA lab, but where Diane Havlier, an AIDS researcher with close ties to the Latinx community in the Bay Area, recognized that that community was being disproportionately affected by COVID. And she had a relationship with what's called the Latino Task Force, a grassroots community organization that had been dealing with Latinx health. So this is an underserved group. It's often undocumented, hard to get to, but through our relationship with Diane and her relationship with Latino Task Force, we were able to set up a program in San Francisco that provided testing for Latinx workers, resources for those who needed it, and then ultimately now vaccination to help understand and slow down the spread of the virus. And this was really something where we learned very quickly, in fact, that this was at a point that literally none of the white or Asian people in the mission we tested, which was over half of all households, were positive for COVID. 5% of the Latinx community was already positive. So reaching out through different people, again, as the IHCC does, was really important to this work. Next slide. Now, having this emphasis then also helped us to build capacity for pathogen sequencing. The project I told you about was a local project. It was in the San Francisco Bay Area. But from another set of relationships and partnerships, CZI had been working on developing capacity for testing for infectious disease worldwide. And the relationships here, again, were the tech group at CZI, the BioHub's experimental biology infectious disease group, and the Bill and Melinda Gates Foundation, which set up a global challenges in order to send genomics capacity throughout the global South. They had 10 partner sites at places ranging from Brazil to South Africa to Bangladesh. And we set up a partnership so that the BioHub trained them in wet lab capacity so that they could do metagenomic sequencing, and CZI trained them in bioinformatics and on a set of software programs that allowed them to interpret that. And this was being used in an agnostic way to find new infectious agents. For example, a chikungunya virus outbreak in Bangladesh that caused an unexpected outbreak of pediatric meningitis. But when the SARS-CoV-2 epidemic hit, these groups were able to rapidly pivot to look for pathogen sequencing of SARS-CoV-2, and indeed to track the virus and to track variants of the virus throughout these different sites. So at this point, we are operating in 15 countries through this starting partnership, which set us up with 10 countries, and then now with additional countries through the Africa CDC. And one of the lessons from this really is that we know that the cost of sequencing has dropped to millionfold over the past few years. And what that means is that sequencing is no longer a luxury. Sequencing is a commodity, and it can be available to all, including those in low and middle income countries. And that's what we're trying to build out here and stabilize for future work on this virus and on many others. In addition, we learned through this work that not all low resource settings are in low and middle income countries. And indeed, the departments of public health right here in the US are low resource settings, settings that did not have the capacity for modern sequencing and modern genetics. And as part of the learning that we developed from this work on pathogen sequencing, we developed a product for the California Departments of Public Health called Aspen, that's allowing them now to do their own direct sequencing, interpret their results, and understand the spread of the virus through their communities and gain actionable insights that allow them to interact with public health. So again, partnerships, starting with different groups, each with their own expertise, and working together to build capacity that did not exist before. Next slide, please. Another one of our flagship areas has been open science, and the pandemic has been a tremendous success for the principles of open science and of sharing information as quickly as possible and responsible. CZI has been a long-term supporter of the preprint movement that included in this past year setting up a new preprint server, medRxiv, to supplement bioRxiv, which we had already been supporting, specifically in medical work. And in the course of the SARS epidemic, there's been a huge uptake in preprints as a way of scientific communication, of accelerating communication, with 25% of all papers first appearing as a preprint in one or another server. Now, we were all holding our breath about whether this would lead to some unexpected problems, but the response has been very favorable overall. We also, for example, share protocols in the SARS-CoV-2 testing community through an open site called protocols.io, and trying to build out these capacities, again, as a way that we feel that we can accelerate science, which has happened so beautifully this year. Next slide. Another one of our flagship projects is in single cell biology, defining and describing the parts list of the human body. Just in this past decade, methods have become available that make it possible to sequence the RNA in single cells and determine their patterns of gene expression. And this has led to an international project, not unlike the IHCC, called the Human Cell Atlas, a global effort to map all 37 trillion cells in the human body as a resource for studies of health and disease. And again, this meets our systemic goals. This is a way of studying all diseases because we can study the cellular basis of any disease through this project that we're funding. In addition, we're both funding and building tools for interacting with single cell biology. Next slide, please. Once the COVID outbreak occurred, we realized that these resources could be highly useful to people who are trying to understand this complex and confusing disease. And indeed, the data that we already had from healthy donors, which we put together on an open website together with the Wellcome Sanger Institute, enabled us to identify very rapidly the cells that expressed the receptors that allowed SARS-CoV-2 virus to penetrate. And this helped to untangle, for example, the fact that the brain effects that people were seeing were probably vascular rather than neurological in origin. So that was an amazing validation of the idea that single cell biology could be useful for studying any disease. In this case, a disease that literally was unknown at the time that the data were generated for the healthy resource. We then started to work with patient donors and realized very rapidly where our projects had fallen short, because the healthy donors we had were universally in middle life. They were 80% Europeans, and yet the patients that we were seeing were Black, Latino, very old, or very young children with various kinds of immune consequences of the virus. And so that made us realize that we needed to rethink our references. Next slide, please. That in fact, reference data should be representative. It should be representative of the broader community that you want to serve. And that is, again, something that the IHCC has taken very much to heart. So today we have a closing RFA specifically to enrich the Human Cell Atlas with a broader network of human subjects representing different ancestries. And also we're currently reviewing a set of grants that are addressing pediatrics and younger patients to understand the cells in their bodies and provide a resource for those studies. Next slide, please. And in a second project based in single-cell biology that we're just launching, we're working on single-cell RNA sequencing of blood cells from multi-ethnic cohorts. This is a partnership with the Wellcome Trust and the Wellcome Sanger Institute, as well as a number of other institutes. We're going to be pilot testing to see if the blood samples that have been collected for the UK Biobank are suitable for single-cell RNA profiling, and that this form of data can be added to the very rich data in these cohort studies. Now, just because we started thinking by this time already about how representation was important in our work, we decided to not just work with the UK Biobank, but rather to also work with the East London Genes and Health Consortium that serves underserved Bangladeshi and Pakistani communities in the UK to sort of have a broader idea about representation from the beginning. Next slide, please. Our other work is not purely in single-cell biology. I want to call out here that in all of medicine, imaging and microscopy are important methods for moving science forward. We've been studying imaging for a while, both by supporting frontiers projects, but also by supporting networks such as global bioimaging. And we are just opening today an RFA to expand global access to bioimaging, whether that's medical imaging, pathology, microscopy for science, but to make that available to scientists in Africa, Latin America, and the Caribbean, and former Soviet countries. Next slide. So these are all aiming toward a new view of what we're doing, not only toward all diseases, but really in all people, of thinking about diversity, equity, and inclusion in the science that we're doing. So diversity, we have some focused problems that we're studying in the US and in California, for example, diversity of the student body in science. But most importantly, we're taking an equity lens to all of our work, US and international. We want to cure all diseases in all people, and we need to focus our work on the people with the greatest unmet needs. And we also want to be inclusive by including many different partners and grantees from a wide variety of expertise areas and fields and persons. Next slide, please. So finally, we're trying to set up a different kind of philanthropy. We have to have a slightly different measure of success. We believe in peer-reviewed publications, but we also think that success is measured by rapid sharing in preprints, by software, data sets, protocols, resources. We'd be happy to fund people who have never published a paper if they're contributing to these kinds of shared resources. We believe in reach and in sharing, and we believe that collaborative contributions themselves are worth rewarding, and therefore we are very supportive of groups like the IHCC that are trying to move collaboration and a new culture to science forward. Thank you very much. Next slide. We're at the end, and I appreciate the time and the opportunity to share our work with you. I look forward to hearing from any of you. We're easy to reach on the web. I look for your ideas and your outlooks.
Thanks so much, Corey. That was fantastic. And if we were in an auditorium, you'd be hearing a lot of applause right now. So I hope you can think about that. I also want to thank you for your kind words about the IHCC and also for sharing such inspirational work that the CZI is doing, particularly in infectious disease, which we're all obsessed with right now. I also just wanted to say in closing that I really appreciate the synergies that you highlighted between what CZI is doing, what the IHCC aspires to do, both in terms of collaborative and partnership types of work, accelerating science, maybe changing the culture of science, and really trying to address the issues around underrepresented populations. So I think we're in sync there, and really appreciate the efforts that you're doing. And again, thank you for your support and for being here today.
Thank you.
And with that, I thank you again, Corey, and I'm going to turn the session over to our session chair for this morning, Adam Butterworth from the University of Cambridge in the UK.
So good morning, good afternoon, good evening, everyone. Fantastic to see so many attendees, so many people from around the world, from different time zones, different cohorts, different continents joining us today. My name is Adam Butterworth. I'm a reader in molecular epidemiology at the University of Cambridge, and for the IHCC, I'm one of the co-chairs of the Scientific Strategy and Cohort Enhancement Group, and therefore it's a great privilege to chair today's sessions. So what we're going to do over the course of the rest of the day, we have two sessions which are going to highlight the progress that IHCC has made in scientific projects this year. We're going to start, first of all, with 5 speakers in this session, starting with the work that IHCC has been doing around COVID-19. We're going to take a break in the middle, and then after that, we'll continue with a couple more of the scientific projects. And then we're going to hear from the some of the working group co-chairs. That's from the data and data atlas side, from the scientific working group, and from the policy group, about some of the progress that each of those working groups have made over the last year. And then we'll wrap up at the end of the day with a kind of summary of what we've achieved over the last year. So I'm going to introduce all of the speakers now, and we're going to run through the talks. Each talk will be up to 15 minutes. There isn't a formal Q&A during this session due to time and logistics, but feel free to pop any questions that you have in the chat, and we'll be able to relay those to the speakers to follow up on afterwards. So it's great to hear about some of the pilot projects that were funded last year by the IHCC through funders such as NIH and Wellcome Trust, starting with our COVID pilot projects. So I'm delighted to welcome, first of all, we'll have Professor John Chambers, who is a Chief Scientific Officer at Precision Health Research in Singapore and Professor of Cardiovascular Epidemiology at Imperial College London. He's going to talk about the project on COVID-19 in South Asian communities. We'll follow that with Professor Michelle Ramsey, who's a professor of human genetics at the University of Witwatersrand, and she's going to tell us about their work on novel coronavirus host susceptibility in South Africa. And finally, for COVID-19, we have Dr. Sarah Bauermeister, senior scientist and data manager at the University of Oxford, and she's going to tell us about the project around the global mental health impact of the pandemic. We're then going to switch gears slightly with Dr. David Hughes, who's assistant professor in cancer epidemiology at the International Agency of Research in Cancer in Lyon in France, thinking about genetically determined BMI and colorectal cancer. And finally, we're going to finish this session with Dr. Hilary Robbins, who's a scientist in the Genomic Epidemiology Group at the International Agency for Research on Cancer, who's presenting about the Opioid Cohort Consortium. So thank you very much to all of the speakers. Ida's going to keep an eye on the time for us to make sure that we don't overrun. If we could have the next slide, please, and hand over to Professor Chambers. Thank you.
Hi, thank you, Adam, very much for the warm and generous introduction. So it's a pleasure to talk about our work that has tried over the last 12 months and that plans over the next 12 months to bring new insights into the impact of COVID-19 in South Asian communities. Next slide. My background, as Adam alludes, is actually in cardiovascular epidemiology. The COVID epidemic, like many of us, took us by surprise, and has been an opportunity for many of us to reshape our scientific strategies. So historically, traditionally, my group is interested in non-communicable disease in South Asian populations, trying to understand the behavioral environmental factors that determine these, and to try to translate the knowledge that we generate about disease etiology into new strategies for prevention. And the need for good population health research in South Asian populations is, for chronic disease, very clear. For example, on the right-hand side, we have the incidence of diabetes over 10 years in Asian populations in blue compared with European populations in red, stratified by age and by gender. And there's a tremendous increase in risk of diabetes, cardiovascular disease, and other chronic disease in South Asian populations that remains unexplained. So driven by the unexplained excess of chronic disease in South Asian populations, my group have put together a network of like-minded researchers in South Asian countries to try to put together a longitudinal population study, a surveillance study, if you like, try to better understand the predictors and determinants of chronic disease. And we try to model our approach on best practices in European and US studies by adopting intensive phenotypic characterization questionnaires, measurements of lung function, accelerometry, and so forth, all integrated into a nice cloud-based data collection platform and straddling approximately 200 sites across South Asia, Bangladesh, Pakistan, Sri Lanka, and India, with 5 coordinating hubs. Next, and this was funded by NIHR and Wellcome Trust, predominantly NIHR. So we started this longitudinal study of South Asians we were doing the pilot work at the back end of 2018 and getting stuck into it in earnest in 2019, so that by the end of 2019, we'd recruited the best part of 50,000 people to the study, ultimate target 150,000. And then we hit this brick wall in March 2020 with the onset of the COVID pandemic and the introduction of national lockdowns that persisted 6 months. Some release of the national lockdowns around the end of 2020, October, November, December 2020, allowing us to restart work, but then followed by upper inset panel courtesy of Johns Hopkins, the tremendous second wave that we've now seen in South Asia, which has then led to a second period of lockdown. Now, at one level, as a non-communicable disease doctor who'd spent a lot of time and effort putting together the processes and mechanisms needed to put together a longitudinal population study of 100,000 people, this was frustrating to see that these lockdowns were presenting a complexity that we hadn't anticipated in terms of project delivery. But click next again, turn it around, and what we have is an ideal opportunity to understand the impact of COVID on a South Asian population, in the sense that we have a large number of individuals with good baseline characterization, biological samples collected immediately prior in the 12 months leading up to the pandemic. Followed by phase 1, we're able to study the same individuals, reassess them during a period of lockdown when actually the disease burden is quite low, followed then by a second phase during which actually the incidence of the disease is high, and we can now start to look at the biological and social and environmental determinants of COVID incidence in South Asian communities. So we can turn the cloud into at least some form of silver lining. So what did we do in phase 1? This was work completed last year. In June 2020, supported by IHCC and by Wellcome Trust, and with many thanks to Mary De Silva and Terry Manolio, we were able to complete a quick survey of our population cohort to try to understand how many of them were symptomatic with COVID, or what the impact of COVID and the control measures might have been on people's day-to-day lives. It was striking that fewer than 1% of people reported either having had symptoms suggestive of COVID or having even needed to be tested for COVID. And yet the COVID pandemic, even at this stage, was having a tremendous impact on their lives. Top right panel shows the anxiety and depression indices as measured by GAD-2 and PHQ-2, with a high proportion of individuals reporting symptoms of anxiety and depression, particularly, it's too small to read, but particularly amongst people from lower socioeconomic classes. Bottom panel shows the impact on people's livelihoods and well-being. Unemployment rates for people in South Asia shot up from 5% to almost 40%, so a tenfold increase in unemployment rates, with that a reduction in work hours and a halving of people's incomes. So even prior to a substantial number of people becoming infected, the very existence of the pandemic was having a tremendous effect on the lives and financial and personal security of South Asian families. So that was work done during the first wave, when it appeared, even though the numbers of cases in South Asia were relatively modest, and it was Europe and the US that was predominantly leading the way. Now, of course, over the last 3 to 6 months, things have changed, and there's been a second wave in South Asia that has dwarfed anything seen in the US or Europe. Again, data courtesy of Johns Hopkins. Next slide. And with the progression of the pandemic, an increasing appreciation that the burden of the disease was not equal amongst populations. And data from UK Biobank, for example, published at the back end of last year, showing that the risk of developing symptomatic COVID or requiring a COVID test as a proxy measure for symptomatic COVID, being about twofold higher, 1.5 to twofold higher amongst South Asian and Black minority ethnic groups in the UK compared with the European populations. And that excess risk being only partially accounted for by measured social and biological factors. And it's not just the increase, there's not just an increased risk of developing symptomatic COVID, but there's also evidence for an increased risk of COVID-related mortality. So this is now data from the Open Safely platform, which is a fused data set of routinely collected NHS data and mortality data, both from primary and secondary care, that allows interrogation of the incidence of COVID in the study population, which is 22 million UK residents. Allows you to assess the incidence of COVID and also potential predictors based upon things measured in the electronic medical record. And what is measured in many electronic medical records is ethnicity. And based upon these NHS data, South Asian individuals and people of Black heritage were up to twofold higher risk of dying from COVID, and in a way that was not apparently explained by, again, known measured risk factors. So this sets this. So this is potentially very important. Here's a susceptibility in major global populations that's incompletely understood. We have to recognize the potential limitations of the data sources that we're using for this. The first is the UK Biobank. Whilst it's got very comprehensive and complete data, the number of South Asian and minority ethnic groups participants is modest. There's only 7,300 South Asians in the UK Biobank study. And whilst the Open Safely dataset has got 22 million individuals in, it lacks biological samples, there's no genotype information, there's incomplete baseline characterization. Indeed, ethnicity itself is missing for 25% or so of participants. So we have hints of a problem, strong suggestions of a problem, but incomplete data on which to answer, to understand the underlying causation. So next slide. So these observations then inform phase 2 of our work, which has got 3 aims. We aim to define the incidence of COVID-19 and its major complications in South Asian populations, both from South Asia itself and in particular from India, as well as South Asians living in migrant settings in the UK. We aim to use and we aim to carry out that assessment within our existing longitudinal population studies, and providing us with strong baseline and clinical, environmental, behavioral, as well as molecular information that allow us to identify the primary risk factors for both acquiring COVID and also the complications of COVID in South Asian communities. And then to use the data created to try to understand, well, what are the primary determinants of the excess risk? Is it something as simple as adiposity? Is it underlying genetic variation? Or is it environmental exposures relation to an increased occupancy of housing and other socioeconomic background factors? The primary aims for this, the primary endpoints for this effort are primary COVID endpoints are threefold. Total COVID infection based upon a PCR test or confirmed positive COVID serology, severe COVID, requiring hospital admission or resulting in death, or prolonged COVID with symptoms lasting greater than 6 weeks. Next slide. The study, the work that we're carrying out involves four interlinked work strands. Work strand 1 and 2 deliver the outcomes identification. Work stream 1 through clinical follow-up of an estimated predicted 30,000 South Asians from 3 longitudinal population studies, the Lollipop Cohort study, the iHealth T2D Cohort study, and the NIHR Wellcome Trust funded GHRU surveillance study that I alluded to in the introduction. 3 population cohorts together with the best part of 100,000 people in. We hope we'll be able to persuade 30,000 to come back for direct physical reassessment, which will involve a questionnaire, building on previous Wellcome Trust REACT study IHCC templates, as well as a blood sample for serological testing for evidence of either COVID infection, bearing in mind the possibility of a vaccine-related false positive in spike protein antibodies. So direct follow-up of 30,000 people, and in addition, record linkage, which in the UK is through to NHS mortality registries, and in South Asia is to, for example, COVID-19 registries held by ICMR, which has only become possible in the last 2 weeks theoretically. So it's aspirational, but we believe there's a strong possibility that we'll be able to deliver that. Next slide. Work strands 3 and 4 relate to the molecular phenotyping and analysis of data. The molecular phenotyping will include collation of existing molecular data from the cohorts, which includes a reasonable amount of metabolomic and genomic information, but will include consistency, a focused US on 2,500 COVID-19 cases and controls. And the analysis will address the 3 specific aims highlighted at the outset. Next slide. Timelines for this, we've been at this already for 6 months. We've got our IRB applications in place. We're about to start the clinical phase of data collection from June onwards, and we hope to complete that in 3 months. Then we've got 3 months for the molecular assays and 6 months for data analysis, with the whole thing being wrapped up about 12 months from now. Next slide. Funding for this, extremely grateful for the primer support that came from IHCC through the Chan Zuckerberg initiative. That was a pump priming investment that enabled us to get a postdoc who then helped us write a more substantial grant application, get our IRB applications in place, and put the pilot data together that informed an application to MRC and to the DBT in India, who have given us more substantial funding to deliver the large-scale effort that is phase 2, involving 30,000 individuals. So incredibly appreciative of the pump priming funding that IHCC have given that has enabled this work to go ahead in a comprehensive way. Next slide. And of course, here is one person representing a much larger collaboration, and it's very important to recognize this is the work of many individuals, and particularly as we've moved into a phase funded by MRC DBT, and it's important to recognize the major contributions of colleagues from the UK, Paul Elliott, Helen Ward, and Jaspal Kuna from Imperial College, and colleagues from South Asia, Dr. Anjana, Dr. Jha, Malay Murida, Anu Kasturi Ratne, and Khadija Khawaja from Pakistan. Next slide. Very good. That's it. Thank you very much for your attention. We look forward to being able to share results in 12 months' time.
Hello, everybody. So I was just thanking John for this fantastic introduction and for saying that you said 2 things that I think are very important and pertinent to my presentation. And one of them is, making the point that there are ethnic differences in response to SARS-CoV-2 infection. And secondly, that there's a lack of data in many parts of the world. So I'm now going to take you from Asia all the way to Africa. So in Johannesburg at the moment, it's just before 2. And what I'd like to do is just update you on our study that we call COVIGEN SA, which is really a host genomic study trying to understand why COVID-19 manifests differently in different people. So the next slide just shows a basic outline of what I want to talk to you about. Just a little bit again about the factors that affect the outcome of infection, but then what we are doing currently with this project called COVIGEN SA, and then our challenges and next steps. So the next slide just shows us what is going on in the world at the moment. And I'm sure that many of you are watching these statistics from different perspectives. This is from World Odometer. And what I quite like about this is it talks about the active cases and the closed cases. And when we look at the world at large, of the closed cases, about 2% of those result in death. And when we look at the right-hand graph, we can see that, we've had the first wave and then a very big second wave. I don't know how many waves we would call it now, but of course it's manifesting differently in different parts of the world, as we just saw this enormous surge in India. So in South Africa, we've already documented over 1.6 million cases, and of those 55,800 odd have resulted in death. And compared to the 2% worldwide, in South Africa, it's 3.4%. But I think a lack of data means that we still can't say that it's higher or lower or the same, because we don't have enough equivalent data. So in the next slide, I just want to just remind everybody again that there are so many things that affect the outcome of infection. That, it's the viral factors, it's the environmental factors, but in terms of what interests me as a geneticist is really the host factors. And, those are the things that we can't change. So age, sex, and genetic susceptibility. But then the other things, diseases of lifestyle in terms of comorbidities, obesity that was spoken about earlier, and diabetes having a huge impact, and then of course people's behavior, and now understanding the impact of vaccination. So in the next slide is a figure from a preprint that came out very recently, which is really this international host genomics or host genetic factor study. And this is a phenomenal study that covers many parts of the world. They have in their cohort almost 50,000 cases from 46 studies in 19 countries worldwide. But if you just click the button again, what we can see is that in Africa, there are no studies that are currently contributing genetic data. And it is important to study Africa and Africans for many reasons. From a genetic point of view, the fact that they have the greatest genetic diversity of any peoples in the world, but also that the genomic structure is different in terms of patterns of linkage disequilibrium, for example. And we also have a unique environment. So looking at the host genome environment is incredibly pertinent. In the next slide is again a figure from the same paper. I know you can't really see or read that, but it is what we call a Miami plot, which is really looking at genetic association on the top relative to individuals that were hospitalized due to COVID-19. And at the bottom, it's showing genetic association with people who were infected, but many of whom would have been asymptomatic or very mildly affected. And the spikes, of course, are the genetic signals of association with each of those two phenotypes. And just interesting to note that the controls that we use to compare them to are population controls where they didn't necessarily have any information on their COVID status. But what you can see is that in the reflection, many of the signals that came up in the hospitalized cases also came up in those who were not hospitalized. But what we can see is that there are some signals that are unique either to the more severe outcomes or to just being infected with the virus. So essentially, it is important for us to stratify and look at the data from different perspectives and try and understand both what gives people increased susceptibility, but also what might be protective against severe disease. And this is something we would love to do in Africa, but of course we don't have nearly those numbers, although we are hoping to build them up with this particular project. So in the next slide, I want to introduce you to Covigen SA and give you a little bit of an update. So again, I know this isn't visible, but what the purpose of this slide is to give you a sense of how we went about this. Because when we started Covigen SA, we had no funding at all. So we didn't build it on an existing cohort that we were now hoping to leverage for this Covigen study. But what we decided to do was to form partnerships with different academics and people who did have cohorts and who were building up groups of patients and participants and who had decided to add a COVID dimension to their project. So if you just click through to the next one. So this is the core partnership. So I partnered up with a nephrologist actually who works at a hospital associated with Wits University. And she has many contacts and works with many people in our region and could therefore identify suitable partners. So if you click the next button, that points us to a group of studies related to one of our partners. And they have 5 different studies. And for all of them, they've now sort of started collecting information on COVID. And already we have ethics approval for a genetic study nested within their studies and have started recruiting participants. On the next click is the green box, which is another partnership, which is really with a group at Baragwanath Hospital in Soweto, very close to Johannesburg. And this is the group where they have 3 different studies, and they've already shared with us samples for DNA testing. And I'll give you a little bit of more of an update in a minute. Then on the next click is another study, which is called First C, the C standing for COVID, because the first study existed a long time ago and is really about following influenza infections and distributions and prevalence in rural and urban communities in South Africa. So what they've now done is to add the COVID dimension, and that is what we're partnering with them on. And I'll give you a little bit more information on that as well. And then we also have, I think, the final button for this slide, another group that we're working on who are actually studying pneumonia. And for that particular study, we've just received ethics approval to also save the buffies and not throw them away, but extract DNA for our host genomic study. And then our intention is also to link up with SAPRIN and Cobus Herbst groups and trying to find additional affected individuals to understand progression of disease and how that links to host genetics. So you can click through to the next slide. And this is just an update of the Soweto group. So they have already collected 360 cases, and you can see that 403 of those were symptomatic. The majority of this group was female. One of the studies is actually pregnant women. And of those individuals, a certain number needed supplementary oxygen, and 18 individuals were in intensive care, but none of them required ventilation. And there were 25 deaths among these cases. And interestingly, many pregnant women, because as I said, one of the sub-studies was actually on pregnant women. So I think there's relatively little done about pregnancy and COVID-19. In the next slide, I'm just showing you an update on the First C study. And they already have got some interesting data. So their objective is to understand the burden of SARS-CoV-2 infection in 2 communities, one rural, which is in the north of South Africa in a place called Agincourt, and the other one in an urban setting in Klerksdorp, which is not very far from Johannesburg. And they also want to assess transmission and the dynamics of transmission of infections. So their model is to find, and they have already followed these 100 households since about 2016, where there are at least 5 people per household. And what they've shown following our second wave of infection is that about a third of people were infected, and about 16% was symptomatic, and of those, 14% had been hospitalized. And what they have seen amongst these participants is that 3% of them have actually been reinfected. So on the left bottom hand side, you can just see some of the field conditions there for the study. And then the 2 graphs, the one on the left shows what is happening in a rural setting. Wait, let me just see. Is that rural? Yes. And then on the right is the urban setting. So what you can see in the rural setting is that the first wave was relatively small, but the second wave much larger. And we think this is because it took a while for the virus to actually reach the rural areas. Whereas if we look in the urban area, the first wave was larger than the second, which may be smaller because there was greater awareness and more preventative measures taken. So in the next slide, I just wanted to update you on the genomics for the COVIGen SA study. And this is the part where we've had our first funding from the IHCC, which allowed us to now have 576 DNA samples genotyped. So I'm very happy to give you an update from last year, which is that those samples should be genotyped in about 4 to 6 weeks. So we're really looking forward to getting those first initial data to start looking at it. And even though it is a small sample size, we hope that we will be able to see some of the high effect associations. I'm happy to report that because we already have some funding, we were able to leverage additional funding from the ALIVE Consortium at our university, which will allow us to genotype more individuals because we know that size does matter when it comes to genetic association studies. Now, fortunately, we don't need to find population controls because leveraging our ABIGEN cohort from H3 Africa, we already have genotyping data from about 5,000 South Africans, and those will act as our controls. So in the next slide, I'm just going to talk briefly about the challenges and the next steps. I think for everybody who does large cohort work, this is sort of the run-of-the-mill challenges that one has. So for us, particularly integrating our phenotype data that we're getting from all the different studies has been a challenge because they've all collected different information. And we've kind of drilled it down to some of the core things that we hope to include in our analyses. We then also will be working with our partners to increase the number of severe cases, because those are the ones that we would like to genotype next, to look particularly at genetic associations related to severe disease. And then, as always, resources and putting those all together is a challenge. In terms of next steps, right at the moment, we're focusing with our partners on assisting with the field work. We've got ethics approval now from all of those groups for genetic studies, and we'll now be working with them to make sure that we get more participants into the COVIGen SA study. We're really looking forward to doing the analysis of our preliminary data, which we hope to have in 4 to 6 weeks, and to really assess and see whether what we're seeing in Africans is the same or different or similar to what is found in the rest of the world, which is primarily premised on European populations. And then, of course, we hope with time to be partnering with other host genomic studies to enhance a sort of sense of what is happening in the world, not only in the better studied parts of the world. So the last slide just shows the core team of COVIGen SA. My partner in crime, June Fabian, And then Andrew May, who's a postdoctoral fellow who spends about 40% of his time on this project. Then Harriet Etheridge, who is a bioethicist and has helped us with getting ethics approval for all these different studies. And then Heather Seymour, who is a PhD student who has just started this year, and this is her main PhD project. I always feel that's a good thing because that moves the project along. And then Heather Maher, who is a nurse and who assists with a lot of the clinical and patient or participant-related work. And that's all from me for now. Thank you very much, and thank you again to the IHCC for the funding for this project, which made it possible for us to get going. Thank you.
So, hello everybody, and it looks like it's me up next. So I am Sarah Baumeister. I am a senior scientist at the University of Oxford and a senior data manager for Dementia's Platform UK at the University of Oxford. I have a dual role there, and here I am PI for the Oxford site of a project which has been funded by the IHCC. So if I can have the next slide, please. So I just want to thank the IHCC very much for both inviting me here today to present on our project, but also for funding this project, without whom obviously the project would not have gone ahead, and our funders, both the Wellcome Trust and the NIH, for this generous contribution. And if I can have the next slide, please. So I'm going to do a very brief overview of what we've achieved in the first 6 months of this project, and how we're going to actually spend the following 6 months of trying to push in as much as possible, but also how we can extend this work into the future, because this is very much a pilot work. And if I can have the next slide, please. I'm going to start with acknowledgements because I am very much a representation of this group. There are 10 of us situated across 3 sites: Oxford, Sao Paulo in Brazil, and Massachusetts at Harvard Medical School. I'm one of 3 PIs, along with Andre Brunoni, Jordan Smoller, and our wonderful group of analysts and data scientists, Delia and Josh in Oxford, Daniel in Sao Paulo, Ashley, Rebecca, Heather, and Liu Zhaowen in Harvard. And this is very much a team effort, working under difficult circumstances across multiple time zones, and all contributing to this project. We are very much a team. Next slide, please. So the project overview. The COVID-19 pandemic has brought an unprecedented set of challenges, as we all know, impacting the mental health of populations around the world. The IHCC COVID Mental Health and Behavioral Impact Scientific Working Group is leveraging the unique platform of the IHCC Consortium to address pressing questions related to COVID-19 mental health. And the 3 PIs, myself included, we are the working group co-chairs for the mental health working group. So harnessing the power of 12 plus, and I say plus because we do have another couple which are pending, cohorts equating to over 14 million individuals, this 3-site project aims to catalogue and categorize these cohorts according to purpose, to harmonize a set of domains and variables for cross-cohort investigations, and to conduct analyses to address core scientific questions. Next slide, please. Our data corpus. Now, this table reflects 6 months of work, and I really think it does an inadequate job to reflect the very hard work of the team. But nonetheless, this was the best way that I could do this, and I hope the team forgives me for condensing their very hard 6 months of work into one simple table. But I'm doing the best job I can here, and I will talk you all through it. So these cohorts represent us approaching cohorts, appropriate cohorts, having a look at the data, deciding whether it was appropriate for this scientific project, cohorts contacting us back again and really being very willing to participate in this project. What started out as with us identifying perhaps 3 or 4 appropriate cohorts, other cohorts saying, please, we really want to take part in this very valuable project. Us going through the variables, understanding whether they had appropriate variables for us, and then others coming on board. And so this process really did take a large amount of time over the last few months. And some of the criteria for participation was obviously we needed longitudinal data, or we needed a cohort to have had at least one wave of data, but had administered, in addition to that single wave, also a COVID questionnaire. That was another requirement. These, you will notice, are IHCC cohorts, but also cohorts from Dementia's Platform UK. DP UK is one of the IHCC affiliate members, and as such, as Senior Data Manager, I do have an inroad into our cohorts, and I was able to approach the appropriate cohorts as well. We are also using the DP UK platform as an infrastructure for the shared analyses of some of these cohorts. So just briefly going down these cohorts, you can see that all of us cohort, we are able to access that in situ at Harvard University. The analysts there at the moment are busy categorizing and harmonizing those data using machine learning and artificial intelligence algorithms, working very hard on that. The Brazilian high-risk cohort have just come on board. We have yet to categorize and have a look at their data to fill in this table. ELSA Brazil are already on board, and we've begun the preliminary work, as with ELSA UK as well. FinGen have just come on board saying that they are very happy to participate in this project. Generation Scotland, UK Biobank, DP UK cohorts, they're on board. They're uploaded onto the DP UK data portal. HRS has just been uploaded as well. And the Oxwell School Survey, thanks to the University of Oxford Department of Psychiatry, they are allowing us to use this unique study for us for this project. I also want to acknowledge the COVID-19 social study, PI Daisy Fancourt, who is going to collaborate with us with this unique study of lockdown and COVID-19 in the UK. We will be working with them to see an overlap of study aims and ideals. So moving on to the next slide, please. Variables of interest. This is another huge piece of work. We had to go through these cohorts and look at the scientific questions we would like to answer, and then look, what do we focus on? It's a bit like a candy shop. We can't look at everything. We need to focus on mental health, and across the 3 sites, think about our strengths. So in Oxford, we want to focus on cognition and mental health. In Brazil, they already have undertaken a lot of fantastic work on mental health trajectories. And then in Harvard, looking at lockdown and the effect and socioeconomic status on lockdown. So we have recognized that these variables of interest across outcomes, demographics, external health factors, social distancing, lifestyle, social support, resilience, discrimination, psychomedical factors, and healthcare access were our focal points of variables of interest. And the team, the data scientist and the analyst, and all of them have come together to categorize these into spreadsheets, and this work is ongoing to search out variables to build data sets towards select harmonization work as well. Core questions, the important part, the scientific part. So what are the core questions that we would like answered? We would like to look at mental health trajectories pre and post-pandemic, socio-demographic determinants of mental health outcomes of COVID-19, neuropsychobiological manifestations of COVID-19, COVID-19 mental health and cognitive resilience. What makes someone able to be resilient to the mental health impacts, and what makes someone not be able to cope with that impact, and the global mental health impact of lockdown and regional restrictions. So some of the preliminary work which is being undertaken over the last month and will take over the next 6 months, in addition to the categorization work which will be ongoing, our colleagues Daniel Fattori and Andre Brunoni in Brazil are working with ELSA Brazil to identify groups of depression trajectories during the COVID-19 pandemic. They're going to be rolling this out across different cohorts, but building on existing work, they're going to be looking at group-based trajectory modeling, looking at the pre-pandemic presence of mental disorder, exposure perhaps to violence, history of trauma, loneliness, and COVID diagnosis. So what they've done here is looking at this initial output, they're aiming to identify groups of depression and anxiety trajectories during the pandemic. So inserting data from the PHQ-2 and GAD-2, they've assessed 3 time points during 2020 in a group-based multi-trajectory model. So 4 groups of trajectory were found, such as low symptoms, moderate symptoms, and high stable symptoms of depression, with and high decreasing symptoms of anxiety. Next slide, please. And furthermore, they've also aimed at investigating the role of pre-pandemic and pandemic characteristics as predictors of trajectory. And what they found was having a diagnosis of mental disorder in 2018 was predictive, as well as symptoms of COVID during the pandemic and concerns about family income and self-report of physical health problems during the pandemic are also predictive. Other preliminary work which is underway in Oxford is looking at early adversity and long-term effect mediated by COVID-19. This also builds on work which we undertake in Oxford, looking at early adversity and dementia. We were able to simply hit the ground running, almost. Makes it sound very easy. It's not. So look, using UK Biobank, ELSA UK, and extending it later to ELSA Brazil, we're going to assess the associations between early life adversity and later life biopsychosocial outcomes, cognition, and dementia. So using structural equation modeling this time as an approach. If we go to the next slide, please. Our early work using UK Biobank and ELSA as examples, I don't expect you to read this, I will just gabble on. We look at childhood adversity as a latent construct, child abuse such as physical, sexual, emotional, and deprivation, and cross-sectional work looking at biomedical outcomes and cognition, controlling for age and income and education. We found very significant effects of early adversity in retrospective reflection of child adversity. And if we now look at the next slide, please, our aim is now to partition this, so looking at lifestyle, mental health, socioeconomics, but this time inserting COVID-19 into that as a mediation effect. So what happens to someone that has, if we look at it in this way, a cumulative neurobiological load, they already have this lifetime stressor, along comes COVID-19, what happens to cognition post-pandemic? So for ELSA UK and ELSA Brazil and UK Biobank, they are collecting data through the pandemic, post-pandemic. We are able to assess cognition pre and post and see how lifestyle and mental health and cognition are interrelated with COVID as a mediator. Next slide, please. Further work which we are undertaking in Oxford is the associations of early adversity, again on brain structures. So this is a multimodal analysis that we're looking at. And this time we've recently published on this work, the associations, again, perceived adverse lifetime experiences with brain structure. What we're looking at is how cortical structure is changing because of the experience of having COVID-19. So if we go on to the next slide, please. Retrospective reports are related to emotional abuse are actually associated with small reductions in the cerebellum and ventral striatum. Our paper is published, you can go and read that. So what we're going to do is rerun our analyses, but then look at people who have experienced COVID and COVID outcomes. and how did they actually fare with a COVID outcome? So either mortality or a very hospitalization, for example. And this is childhood adversity experience before the age of 16. And we're going to repeat this and see what is actually happening. So UK Biobank are actually imaging again, and we're going to take this third wave of imaging, and the percentages shown here show that we have a very good spread of adversity data among both positive and negative COVID cases. So, and also we have the GAD scale as well to be able to use, and also the PHQ to measure depression measurements as well. And what we will also be doing is integrating a genomic measure to look at a multimodal analyses using ApoE as a mediation as well. So this work will be undertaken by Oxford over the next 6 months. So our project has now almost completed cohort confirmation. We have a few cohorts which are yet to confirm their participation in the project, and the cataloguing of variables of interest. We will still be continuing to harmonize a few of these variables amongst our cohorts, because there will be analyses which will benefit from the harmonization of a very small data set. And merging of data sets for select analyses. Not all analyses, such as the ones I've just cited to you, will benefit from harmonization of data sets. For the big analysts of you out there, you know that raw data is often more beneficial for these big analytical procedures. We do appreciate our project is ambitious, but so are we. And as we continue to be challenged during these times, we are very optimistic that our project will contribute towards the understanding the global mental health impact of COVID-19. And most importantly, some of the cortical atrophy work and the cognition work, we hope will feed into the understanding the work of long COVID and what is happening actually to the physiological brain and the long-term impact on cognitive outcomes, where only now the research is coming out where people are mentioning the cognitive impact. And we hope that our preliminary work will likewise feed into funding applications. And we do see this as only a starting point. Next slide, please. And I would just like you to focus on our wonderful cities that we are all representing. And I want to emphasize that this project is we want to see it as a multicultural project where we have representation from South Africa, from Brazil, from Europe, and from the UK. And we really hope that the rich diversity of our cohorts will feed into the outcomes that we are putting into this project. And we will still welcome input. And we do thank our cohorts and the participants extremely from the bottom of our hearts, because without them, this project would not be possible. Thank you very much.
So welcome, everyone. I am David Hughes, so I'm at the University College of Dublin in Ireland. So the title of my project was Exploring the Role of Genetically Determined BMI in Infancy, Childhood, and Early Adulthood on Colorectal Cancer Development in Later Life. As this project only actually just started in beginning of this month in May, so I didn't have a lot of progress to update you on. So I'm just going to summarize a little bit of the background again, and then just some of the initial work on the project. So this project depends on this nexus of contributing etiology to colorectal cancer development through what we term as factors in excess energy. That obesity would be a primary risk factor for this, along with other metabolic dysfunction. And we hypothesize that this early exposure to metabolic dysfunction and obesity at different life stages may differentially impact risk of later colorectal cancer. So we don't know exactly how much obesity in infancy and childhood will influence colorectal cancer development. And most of the assessments is done with the obesity in adulthood. And certainly when we look at genetic risk factors, they're generally they're generally associated with adult onset BMI. So we want to look at different genetic risk factors for throughout the life course, so that we can investigate how the propensity and temporality of genetic predisposition towards common obesity across distinct life stages may differentially impact the risk of adulthood colorectal cancer. So next slide, please. And to do that, we're going to construct 4 genomic risk scores. So we're going to take what's been found in genome-wide association studies for the genetic variants that influence BMI, and we're going to construct those that have been found at different life stages. So from infancy, so we have a genomic risk score for obesity in infancy, then a genomic risk score for childhood obesity, also one for adulthood, and then the overlap between childhood and adulthood gives us 4 genomic risk scores that we will weight by how much the genetic factor is associated with BMI, and then we will explore the association of those genetic risk scores for obesity with development of colorectal cancer later in life in 3 large cohort studies: a European Prospective Investigation of Cancer and Nutrition, EPIC, from GECO, which is a consortium of different colorectal cancer studies from around the world, and also from the UK Biobank. So this just shows that the project has started in May. I'm going to speak a couple of points on that in a moment, and to say that also we would be very happy once we get going to include other cohorts. I was very interested when you had the African cohort meeting a couple of months ago, attending that, and I'm looking forward to the presentations later on that, because we would love to apply our genomic risk scores to other cohorts. So please contact me if you're interested. I'd be very interested to hear from you. So david.hughes@ucd.ie. So the first thing then we're doing right now is the recruitment of postdocs to work on the project. So we have one postdoc to work in this with our co-lead with Mazda Janab at the International Agency for Research on Cancer in Lyon. And I've also got an ad out for a postdoctoral position for 1 year here with us in UCD in Dublin. So that ad is on the UCD website on their job vacancies. So please, if anyone's interested, please apply. It's open to June 2nd. And then the objectives across the timeline with the project are split into constructing the different genomic risk scores. So that'll be our first task. And then to determine whether to what extent they associate with colorectal cancer risk, and then look at different BMI trajectories and age categories. So we're interested to see if there may be a role, for example, in childhood obesity with early onset colorectal cancer, which is something we're seeing an increasing incidence of. And then for the cohorts where we have got metabolic markers, especially EPIC and the UK Biobank, to look at that interaction of the exposome with the metabolic dysfunction and obesity. So the first thing has been the data access. So with Neil Murphy in IARC in Lyon, we have access to the GECO and UK Biobank data. And so Neil's checking on whether we can also have direct access to individual level data or whether we need to make a new application. So he's doing that at the moment. And then Mazda is handling the access to the EPIC data, which is all fine. We have permission, but we just need to get further permission for their biomarker data, and whether indeed we'll just need to apply to the ethics committee. And we will apply, in fact, anyway, to the ethics committee next month in IARC for the entire project. And again, just to reiterate what I said that there's a postdoc position currently open with me, and there's the website for and the reference for that post. If anyone's interested, please have a look. And then in IARC, Dr. Nicholas Papadimitrou is going to work on this project under the supervision of Neil Murphy and Mazda Janab. So Neil has a lot of experience in Mendelian randomization studies. Indeed, Nicholas has been working with him in several of those studies. And he will contribute to constructing the genomic risk scores and assessing the genomic risk scores in colorectal cancer risk associations. And the postdoc here with me will be mainly focused on looking at the metabolic dysfunction and circulating biomarker data. I just wanted to mention as well a little development from this study. Hopefully I'll be recruiting a PhD to work on extension to this study to look at other cancers in addition to colorectal cancer, just the same idea, but for other cancers, and also to look at survival, how the genomic risk scores may impact survival from cancer. So this is part of the Genomics Data Science Consortium in Ireland, which is to train a new generation of genomic data scientists. And the interesting thing about this is that you apply to this program to be accepted to undertake a PhD in all bioinformatics and genomic related projects. Quite a lot of metagenomic projects in this network as well. And if you're accepted onto the program, you have the power because you choose your project. So there'll be about 30 different projects in September, to start in September, from 6 Irish universities, involving 20 partner organizations in Ireland and externally. And you will have a choice as a student to pick your project, to pick your supervisor. So my project on the genomic risk scores is in for selection, but I don't know if someone will actually select it. Hopefully they will, and then there'll be a PhD student starting on this in September, but they have the power of choice. So it's an interesting program in the way that's constructed. Yeah, just a few points. So when I was invited very kindly by the organizers to speak at the meeting again, and very happy to do so, and thank them very much for the funding that's enabled us to begin on this project. So I mentioned, I said to Ida and Hannah, well, I didn't have a lot. We just started in May, so I didn't have very much to present. And they mentioned that, if I had something to say about any points they could make on the grant registration process or whatever, also to bring those up. So I just thought a couple of things that may be helpful for future and negotiations. That we had started a bit later than we planned, and some of that was just the administration of registering the grants with 3 different institutes, which I can imagine is it's a difficult process. And a lot of it seemed to be just delays on simple things, because obviously it was a new funding organization, and then some of the new institutes that they hadn't previously had contact with. So it was a lot of email exchanges. And I think what happened is that each side for a while thought that the other was waiting on the other to finish something or to do or answer on something, and it could be something quite simple. So maybe in future, even just an introductory, I hate to say another Zoom or conference call type meeting, maybe good, but I think an initial one would be good if it's a new institute, and that will obviously improve as the consortium, its funders, and the institutes become more familiar with each other. But it was just like a couple of small things, like on stipulation on IHCC membership, which is, for example, we didn't have the cohorts at UCD, so it took a little while to work through those points. And again, I think hopefully future funding that'll be easier. And then there was just simple administrative things, like, for example, 2 years, I think, is probably a better project span, or at least what year durations, because what we find is, for example, our hiring strictures limit us on what we can offer. So, for example, at UCD, you can't offer a postdoc 18 months. It has to be a full year. So you can offer them a year or 2 years, but you can't hire them for 18 months. So just some points like that. Whereas I've been very happy to have had the postdoc here for 18 months, but it can only be for a year. So just a few little things like that may be enlightening for the future. So just to acknowledge then the co-lead for this project, Mazda Janab, who knows listening in IARC in Lyon, and colleagues Neil Murphy and Heinz Freisling, Nicolas as well, starting on the project, and all our EPIC associated colleagues and our colleagues in the UK Biobank and Gecko, and my collaborator as well on this project, Veronica Federico, who just moved in January from Emory to the MD Anderson in Houston in Texas. And thanks very much, and thanks again to all for the funding and the support.
Hi, everyone. My name is Hilary Robbins, and I'm a scientist at the International Agency for Research on Cancer in Lyon, France. And I'm presenting today on behalf of Maddie Sheik, who is really the brains behind this project, although we're working on it together. Unfortunately, he's sick this week and has lost his voice completely, so you're stuck with me. Next slide, please. So the title of our project is Opioid Cohort Consortium, or OPICO, to investigate the effects of regular opioid use on mortality and on cancer development. As a brief outline, I'll give you an overview of the project and its aims, as well as the current status and the progress we've made in the past few months, which includes a feasibility study. And then I'll tell you about our plans for future funding and one of the challenges that we've faced. As you all know, there is a global crisis of opioid use and misuse that has led to thousands of deaths and billions of dollars in economic losses every year. And although we know quite a bit about the acute and short-term effects of opioid use and misuse, the long-term health consequences are largely unknown and poorly characterized. And in our case, we are specifically interested in potential associations with cancer. There are three primary categories of opioids, which are important to define because we will use them ultimately in our analyses. The first category is natural opioids or opiates, and this includes primarily opium, as well as some natural derivatives of opium. The second category is semi-synthetic opioids, and these are synthesized in laboratories using natural opioids, such as opium. And finally, synthetic opioids are those that are fully synthetic, synthesized in laboratories, but without using natural opioids, although they have the same chemical structures in order to mimic the same effects. The schematic here gives you sort of the lay of the land in terms of what's known scientifically about links between opioid use and cancer development. So on the left side is evidence for opiates, mainly opium, and on the right side is for prescription opioids. So with respect to opium, there have been a few high-quality human studies, which included 2 cohort studies and 30 case control studies. And these suggested associations between opium use and cancer at 9 different sites. There is also evidence from experimental studies which suggest genotoxic and carcinogenic effects. And this ultimately led to opium consumption being classified as a group 1 carcinogen by the IARC monographs just in the fall of last year in 2020. So this was quite recent. But this was on the basis of evidence for lung cancer, larynx cancer, and bladder cancer. So for some other cancer types, the evidence is still considered insufficient or unclear. For prescription opioids, the evidence from human studies is much weaker because in large part it has come from registry data linkage studies, which don't usually include information on important confounders such as smoking, alcohol, chronic health conditions, etc. Although the data that does exist suggests potentially some of the same associations with the same cancer types as for opium. And again, from the experimental studies, there is evidence for similar effects. So it was really Maddie who noticed this really important gap in the literature and saw an opportunity to try to address it with a cohort consortium approach. The limitations of the current evidence are, again, the lack of ability to address confounding and biases, and the fact that even if you were to analyze a cohort study that had information on those confounders, there is typically not a sufficient number of opioid users to say anything statistically robust. And so the opportunity here is to pool combined data from multiple different cohorts that have information on opioid use as well as confounders in order to get a clear answer to the question of whether opioid use is associated with cancer, specifically prescription opioids, and to better clarify the relationship between opium and cancer. So with that in mind, we founded the Opioid Cohort Consortium, or OPICO, and we were very fortunate to receive pilot funding from the IHCC with the overarching goal to build a strong international resource for multidisciplinary scientific studies on the use of opioids and their long-term effects. We are evaluating the effects of natural opioids, so mostly opium, which we've assessed, which the cohorts have assessed using questionnaires. And for the prescription opioids, this exposure information comes either from medication questionnaires or from linkage to national medication dispensing records, which I'll talk a bit more about in a minute. The main outcomes we are interested in are cancer incidence, as well as cancer mortality and mortality from all causes. In broad strokes, we will organize data on opioid use from different prospective cohorts. And for prospective cohorts that have been linked to national medication dispensing records, we will use these linked data to define the exposure of regular opioid use. Once this is all harmonized and compiled, we will assess the type, distribution, and extent of opioid use across diverse populations and determine the association of opioid use with cancer incidence and mortality. As of now, there are 10 different cohorts that have agreed to join the OPICO. These include 2 cohorts from the USA, 4 cohorts from Europe, including France, Scotland, UK, and potentially some other countries from EPIC, 3 cohorts from Iran, as well as 1 cohort from Australia. All in all, there are more than 1.2 million participants in all of these cohorts combined, among which we estimate there are more than 113,000 participants who regularly used opioids. So this is really an important amount of statistical power that we'll have to address kind of these unanswered questions. As I mentioned, some of the cohorts have gathered opioid exposure information from questionnaires, whereas others have done data linkages. Since October 2020, which is when we were awarded the IHCC funding, we have obtained central ethical approval from the IARC Ethics Committee, as well as approvals from 4 local committees for the participating cohorts. And we've submitted 7 formal applications for data use to the different cohorts. And 2 new cohorts joined OPICO. These were the Women's Health Initiative from the US and the Constance cohort from France. And scientifically, really the main thing we've been doing, in particular Maddie, is working on the protocols for harmonizing the data. And he's been doing this in conjunction with different members of our team, including pharmacoepidemiologists, representatives from the individual cohort, cancer epidemiologists, clinicians, statistician, and a data manager, to try to make sure that we kind of cover all the bases regarding the different issues that are going to come up when we work to harmonize these data. And I'll talk more about some of the specific issues. Next slide, please. So something that was quite important for us to do was to demonstrate that it is feasible to define the exposure of regular opioid use using medication data that has been linked to participants from prospective cohorts. So in our case, the cohorts participating in OPICO have already done the linkages. So it's a matter of basically reanalyzing the medication use data in order to assign exposure variables to each of the participants in the cohort. So we analyzed data from the Australian 45 and up study in order to demonstrate the feasibility of this. And we did this together with collaborators at Cancer Council New South Wales and the University of New South Wales, Sydney. These included Kieran Canfell, Mariana Weber, Peter Sayrich, and Sally Pearson, they've really been an incredible group of collaborators to help us understand exactly what we're getting into. The 45 and up study recruited originally about 270,000 adults between 2006 and 2009 from the general population of New South Wales. And the cohort was subsequently linked to the Pharmaceutical Benefits Scheme, which is Australia's national drug subsidy program. So one of the key considerations when we're trying to use these linked data to define opioid exposure is to choose a time frame during which we are trying to estimate whether someone was exposed to opioids or not. And some of the considerations we have in mind when choosing this time frame are the ultimate need to harmonize the linkage-based data with the other cohorts that have used questionnaire-based data, and to minimize possible misclassification in defining the exposure of opioid use. While the questionnaire-based cohorts typically have information about opioid use anytime during the participant's lifetime, the linkage-based cohorts, of course, are only providing medication dispensing records for a specific period of time in that individual's lifespan. So it becomes a bit challenging to really make these two exposures happen in the same time frame. So taking into consideration all of these needs for the 45 and up study, what we ultimately decided to do was define opioid exposure as occurring anywhere from 12 months before recruitment to 1 month after recruitment into 45 and up. So another important consideration is how the policy of medication dispensing in the country and the subsidy programs and the pricing of the opioids at the time of the cohort recruitment all feed into whether the information on medication dispensing was recorded or not. So in 45 and up specifically, just to give you an example, there were 2 different classifications that individuals could be in with regard to their subsidies for medications. And some individuals had a copay of $5 and other individuals had a copay of $35, $32, I believe. And at the time of recruitment into 45 and up, the price of opioid medications was usually between $20 and $25. So that meant that some people got a subsidy because they were only going to pay $5, and the other people did not need a subsidy because their copay was $32. And the PBS scheme only recorded the medication dispensing information if a subsidy was made, which meant that only the recipients of who were in the category of $5 copay would have been recorded when they had opioid dispensing. So ultimately, it was these particular beneficiaries that we had to restrict to in order to do our analysis in 45 and up, because for the other participants, they weren't receiving subsidies for opioid medication, which meant that it wasn't recorded whether they had received opioid medication or not. So from this cohort of about 270,000, I think there were about 37,000 individuals for whom we could actually define the exposure. And among those, I believe something like 8,000 were ultimately classified as opioid users. So again, what we have to do is try to minimize possible misclassification. And sometimes what that means is identifying additional inclusion and exclusion criteria before we can pool the data and analyze the cohorts. So that's just a flavor of some of the issues we've been tackling over the past few months. But all in all, it's been quite positive. And we do feel that it's going to be very much feasible to pull a large portion of the data and ultimately get more or less correct answers to the exposure definition and the links with cancer, as long as we really do our homework in terms of understanding how the data were generated in each specific setting. So moving forward, one of our key priorities, of course, is to obtain more long-term funding. So we will submit an R01 application to the US NCI, hopefully at the end of next week. And then after that, we plan to apply for funding, likely from more local funders, such as the French National Cancer Institute. And finally, one challenge that's interesting to highlight that we anticipated to some degree, but have been able to understand in more detail recently, is that some of the cohorts included will likely not be able to send their linked data directly to IARC because of regulations like GDPR. But it's not only GDPR, it's also a strict data protection regulation in Australia. And anytime there is a linkage between the cohort and national medication dispensing, it becomes even more difficult to do any physical data transfers. So what we plan to do is use a sort of a hybrid model where we pull data from as many cohorts as we can, but also use an additional distributed analysis model for those that we cannot pool. And meta-analyze the results at the end. So we anticipate in particular needing to do this for the 45 and up study, and we will try to avoid it for the others, although some of these issues are still pending. So we do still invite any cohorts that are interested in joining OPICO to contact us. If your cohort is interested in joining, the key data requirements are listed here. In particular, of course, you have to have information on opioid use. This could come either from questionnaires or from linkage to medication dispensing records. And of course, the important confounders include smoking, alcohol, chronic health conditions, etc. And you can contact myself or Maddie. And Maddie also told me to mention that he uploaded some of the key information about OPICO, including the contact information, to the conference portal, so you can access it there. And finally, I want to say a very big thank you to all of our collaborators around the world from the participating cohorts in OPICO. Some of these people have worked very closely with us over the past few months, and we've learned a lot from them about how to make this project successful, and we're quite excited to be working with them in the coming years. Thanks so much.
Fantastic. What an incredible set of talks. Amazing to hear the progress that's been made over the last year, and perhaps even more amazing that all of our speakers have stuck wonderfully to time. So we're bang on schedule. As the slide shows, we're going to reconvene in about half an hour at 20 past the hour, when we'll hear from the remaining pilot projects and hear a bit more from the IHCC working groups about what they've been up to over the last year as well. So go and refresh, get some fresh air, recharge the batteries, and we'll see you again in about 30 minutes. Thanks very much. Great. Well, welcome back, everyone. Hope everybody's feeling refreshed and ready for some more multi-cohort science. As you see from the slide here, we've got another set of talks coming up, 6 speakers. We're going to start by going through some of the progress on the additionally funded pilot projects. So we'll start with Arash Etemadi from the National Cancer Institute, NIH, who's going to talk to us about the metabolomic biomarkers in diverse ancestries project. That will then move to Rhoda Au, who's a professor at Boston University and a senior director of neuropsychology at the Framingham Heart Study. She's going to tell us about the Davos Alzheimer's Collaborative Project. We'll switch to Patrick Sleeman at the Perelman School of Medicine at the University of Pennsylvania, who's going to talk to us about the Polygenic Risk Scores pilot project, both for the IHCC and now for the Davos Alzheimer's Collaborative as well. And we're then going to switch, and we're going to finish the day by hearing from so far 3 IHCC working groups that are in place. So we'll hear from Christina Young from the Ontario Institute for Cancer Research, who's going to talk to us about progress with the Data Interoperability and Infrastructure Working Group. Hakan Hakanarson from CHOP in Philadelphia will talk to us about what's been going on with the Scientific Strategy Working Group. And finally, Laura Rodriguez from the Patient-Centered Outcomes Research Institute is going to talk to us about the Policy and Systems Working Group and progress there. So do feel free to post comments in the chat. They're all being collated, and hopefully people can make connections if you want to follow up with the speakers in the Socio platform. And over to Arash.
Hello, everybody, and I'm very happy to be joining this wonderful meeting. So today I'm going to talk about an update on this project, this multi-cohort project in high-throughput metabolomic biomarker measures in diverse ancestries. So I'm really presenting on behalf of this group of PIs, this great team that have put together this project. Adam, Andre, myself, and Hakan, and also colleagues at CHOP who made this work possible. So as a quick background, as you know, chronic diseases impose a high burden on health system in different countries and all across the world. And it's very important to be able to make early diagnosis and intervention in many of these situations, and health outcomes can be really improved if we can make those. But early diagnosis is often unavailable for, particularly in low resource settings and low middle income countries and minority populations. So it's very important to find ways to promote this kind of early diagnosis, or ways of classifying and stratifying people on the baseline risk. And metabolic profiling is a highly scalable model for risk prediction and prevention. And in many situations, these metabolic profilings have been used in order to categorize people into risk categories. And because it's relatively low cost, it offers a way to individualize medicine for these populations. So it's a great opportunity within the IHCC to try and to implement some of these metabolic profiling methods and put them into test in diverse cohort populations. So this project was started with the aims of with several aims. The first one is to develop the structure for metabolomics, and this is a strategy to develop this strategy for coordinated screening, metabolic profiling, and clinical implementation for all the IHCC. So the major objective of this project was to try to build this infrastructure, this way of actually forming this metabolic platform. Then we, as a second aim, we want to generate the metabolic profile on about 5,000 individuals with different genetic and health outcome data from diverse cohort and population and backgrounds, and analyze the association between different metabolites, such as lipid profile in particular, and genetic and phenotypic data. So in order to do this, we're using the Nightingale blood biomarkers. Many of you might already be familiar with the panel that Nightingale Health is using for the metabolomic profiling. This is an NMR-based platform, nuclear magnetic resonance spectroscopy, which utilizes high technology to detect more than 200 biomarkers. You can see the spectrum of biomarkers on the right hand. It includes lots of lipoprotein particles, fatty acids, subclasses of lipoproteins, glycolysis-related metabolites, amino acids, fatty acids, cholesterol, ketone bodies. So a bunch of inflammation biomarkers. So it entails a lot of important biomarkers related to many diseases, particularly cardiovascular diseases and health outcomes that are very common in different populations. And the good thing, the great thing about this platform is it uses very little amount of plasma or serum, 100 microliters. So with so much little volume of samples, you can measure so many different important biomarkers in this population. This makes it ideal for the use in a larger scale study of different populations with limited resources. Next, please. So there are 4 cohorts that at least at this stage they're taking part in this initiative. The South Asian cohort, includes 1,500 samples of South Asian ancestry from Bangladesh. Adam is the PI for that study, or the lead for that study. ELSA Brazil, 1,000 samples from Brazilian civil servants. Andre is the lead investigator from that project. Golestan cohort study, which I represent, includes 1,000 samples from Northeast Iran, general population from the Middle East, from Iran. And finally, Children's Hospital of Philadelphia, CHOP Hakan is the principal investigator from that project, 1,500 samples from African-American children. So we have both adults and children from different backgrounds and different ancestries involved, and this makes it a truly international collaboration. So although we have developed this project within these 4 initial pilot studies, the intention, and as I mentioned, this is the first aim of this project, is to scale up beyond these 4, and by showing that this is feasible and that the results can be used in different analysis in different cohorts, we want to actually persuade the other cohorts to join this initiative. Other potential places where this platform can be used include UK Biobank, Mexico City study, blood donors cohort, and Kaduri Biobank. These are the different places which we can scale up. We hope to scale up potentially this project to, after showing that it is successfully implemented in these 4 initial populations. Next, please. So I'm going to take a commercial break. So this is an example of how IHCC can help in including different populations, different cohorts. And I invite everybody to tune in tomorrow for a panel on how low middle income countries and low research settings can benefit from IHCC and resource gaps and barriers to this collaborations, which is going to be tomorrow at 14 UTC. 2 p.m. UTC. So I hope you can join that, and we can discuss more examples and more ways of how IHCC can help in bringing cohorts from different populations, particularly low research settings, together. So the cohorts I've mentioned, in addition to the general goal of putting together this metabolomic profile and showing the feasibility of this study with Nightingale, have their own cohort-specific outcomes. The South Asian cohorts aim for diabetes as the outcome for their analysis. We at Golestan Cohort Study would like to see ischemic heart disease. This is a case cohort setting that we have selected these individuals to be analyzed, these 1,000 samples. And CHOP is particularly interested in autoinflammatory disease in children. As I mentioned, these are children of African American ancestry, so they are focusing on that particular outcome as their cohort-specific analysis. And this shows how a global initiative can also have cohort-specific and disease-specific outcomes in mind for each of the cohorts involved. So the timeline, we were very lucky to be able to ship all the samples, all 5,000 samples by the end of December, despite all COVID challenges and all the shipping difficulties going across the globe. And this is also a very nice success story of how these samples from all across the world were shipped to Finland, and they got ready to be analyzed in time. Now we are May 2021, which is right now. We have received word that all samples have been analyzed and the results are ready to be released. So we are hoping, as we speak, we are hoping to receive those results soon from Nightingale. We hope to be able to do project-specific analysis by the end of July, and by September to report back to IHCC and make recommendations as to scaling of the project and different ways of involving other cohorts in the project. And upon completion, of course, data will be made available to IHCC cohort members. And next, please. So for those of you in Iceland and you miss having in-person meetings, so this is a reminder of the last meeting we had. Hope to see everybody again in person. And I want to thank IHCC for funding this project and for giving us this opportunity. Next. And again, tune in tomorrow for the session on LMIC low resource setting cohorts outlook. Thank you very much.
So I wanted to thank the IHCC for allowing me to present today to you. And I want to introduce to you the Davos Alzheimer's Initiative. So dementia and Alzheimer's disease in particular has been recognized by Davos, the World Economic Forum, and many actual international organizations as a really growing worldwide pandemic. And this is really because of the rapidly aging world population. It's actually, for the first time ever, people over the age of 65 now outnumber those under the age of 5. So, but despite this significant shift in the world population, in terms of age demographics, the current limitations of the work that's being done is that 90% of the data in the study of dementia, Alzheimer's disease has actually been inquired on only on people that just represent 10% of the population. So this Davos initiative really seeks to change this. And through the work that we've done just in the last few months, because Davos was really launched in January, end of January of 2021, there have been 3 major work streams that are working together in a synergistic ways. These work streams were actually developed over the course of 2020 with an international group of researchers in this space. So they include not only the global cohort development effort that I'm going to be speaking about more in this particular presentation, and I think also directly relates to the interests of the IHCC. In addition, there is the clinical trials that's developing an international network of Alzheimer's disease clinical trial sites to really accelerate the pace of drug treatment discovery. and validation, and then health systems, which brings together clinical care solutions to the patients and their families. And although we identify 3 separate work streams, we actually work interactively together. So consistent with the objectives of the IHCC, the words of Dr. Bergman earlier this morning in her opening keynote presentation, the global cohort development priority focus is really about creating an even playing field across all resourced environments. So again, consistent with earlier presentations, we understand that low resource environments can happen in countries with low, middle, and high GDP. And so what we're trying to do is strategically figure out how technology actually provides the means to achieve this parity objective. The idea here is not to simply use technology to do a better version of what we're already doing, but rather to harness its power and actually maybe leapfrog over existing infrastructure considerations at the local level. So let me give you an example. How can we use technology to solve the problem that there's a paucity of dementia experts that can actually diagnose disease in many locations? So I always like to point to the Far East, because it turns out there's no PhD programs in neuropsychology in that region, like none. This includes China, which has the largest aging population in the world. And they have no neuropsychologists, and they have to speak of, because they have no formal training programs. So the current practice is actually to export cognitive assessment tools that have been largely developed in the US and Europe. But the problem is that when you do that, these assessment tools are actually fraught with educational and cultural bias. In addition, another problem is, given the paucity of dementia and AD experts in many locations across the globe, many low-resource environments have built their dementia AD studies from recruitment of hospital and our clinic-based patients, which all of you know are highly biased toward any disease of interest and can lead to inappropriate population-level estimates that inform public health strategies. And finally, the other issue is, how do we unleash the opportunities of data science and artificial intelligence at the global level, and really find a way to bring talent wherever it sits to accelerate the pace of discovery? This is particularly important in the realm of Alzheimer's disease, where what we don't know is likely actually still greater than what we do know. And this disparity of knowledge is being further fueled by the homogeneity of the data that's available to date. So if we can go to the next slide. So with this context, the foundational phase of Davos, which is really this year, this current year of 2021, we're actually seeking to build the foundational infrastructure that's needed to push forward a global strategy that will then be able to roll out in the subsequent 5 years. After connecting with a number of global cohort consortia, looking at the work of the IHCC under the leadership of Elias Zerkoni, Hakan, and Jeff, we've actually identified 3 areas of initial focus from which to build a global cohort that will meet the objectives of both global and then also inclusiveness. And so these are really sort of the 3 areas that we're starting at. So if we can go to the next slide. I don't really need to tell you sort of the value of blood, but it turns out that almost any place can collect blood at some level. And so, and clearly, and you'll hear more about some of the plans within the IHCC in the next presentation on how we can take sort of the GWAS and push forward in developing polygenic risk scores in a way that we've never been able to do before in particularly diverse cohorts. In the field of AD, diagnosis remains a challenge, and there's a real movement toward blood-based biomarker data to replace sort of very costly imaging and other gold standard methods like lumbar punctures, removing CSF in order to measure these biomarkers. So this is an area of emerging interest that has great implications for low-cost diagnostic opportunities. And then, you know, it's very important to remember that AD is a very complex disease in which the etiologies and the risk factors are many. And so we have additional clinical risk factors that we've identified, exposome data as well that have been identified as risk factors across the entire lifespan, because AD is now considered a life course disease. And then, of course, the value of blood is that you can store it and use it for future opportunities as science continues to advance. What I really want to emphasize is where I think there is value of digital. So part of our initial plans in this foundational phase is to be able to use digital technologies and really start to phenotype extensively, regardless of what the resource environment is. And we've really centered our solution around the smartphone, because the smartphone is the most penetrating technology in the world. There are currently an estimated 3.55 billion smartphones being in use. And the projections are by 2023, which is really just around the corner, that number will actually more than double to 7.33 billion people having a smartphone. So this is a computer in the palm of everybody's hands, wherever they may be. And you have to harness that. And that is the real power of digital. So with the different kinds of digital applications, you can develop opportunities for self-administration, which you can do on an ongoing basis. And also within a clinic through very minimally trained staff, because a lot of it can be automated in the process. And one of the things that is interesting about a smartphone is that every single one has at least 27 different sensors from which to collect data. And that really opens up the opportunity to do passive monitoring of all sorts of related behaviors, and do it without a lot of burden to the participant. This is a low-cost solution. It's very easy to upgrade to contemporary standards. And it also provides a repository similar to blood, in that as new algorithms, new solutions are developed, you can reprocess this data and bring it up to whatever are today's standards. And it's a non-diluted resource. It doesn't go away. So you can keep reusing it repeatedly without decreasing the actual digital data itself. Some of the results could actually be obtained in real time. And you know, in the world of digital, everything is always upgrading. And that's something that's easy to do immediately and virtually. It can be highly adaptive and personalized. It can provide feedback to users to provide them direct value. So it's not just about us collecting data, it's about providing data that's of use to the participant. And that as we learn more about a participant over time, we can further customize that to their particular needs. And then there are all sorts of innovative opportunities. One of the things that's very important to remember about digital and why it creates such an equal opportunity is that we don't know very much about it yet. We haven't done very much, and there's not much that's been collected to date. So this is really an opportunity to even that playing field across all different cohorts, collect the same base of data, and then start to push it out there to let everybody start to develop solutions with no advantage to high-resourced environments over low-resourced environments, because there's been so little done to date. There's very little precedent. So we can all build together at the same time. And then this is actually aligned with Arash's presentation on metabolomics, where we're really looking at the value of at-home sample kits. So with our colleagues at Duke University, led by Rima Kadora-Doak, we're actually going to start piloting the use of in-home sample collection kits that will collect spot blood as well as fecal samples in order to look at how we can enable things like metabolomics, which you've now heard, is very dynamic as compared to genomics, and allows us to track on an ongoing basis. One of the areas that Davos has been developing is this idea of how do we push forward and accelerate discovery? Because as I mentioned earlier, there's a lot more that we don't know and haven't done than probably what we have. So how do we now harness the data science AI talent and bring it to help solve these problems? And we have many problems. In my world of Alzheimer's disease, there's a lot of can't. So for instance, we can't aggregate data together that's been collected by different groups because the protocols are different. Well, to me, that's just a data challenge. That's about where you take really messy data, you say that there's difference in the data collection protocol, you put it out there to the scientific community, and you say, solve this problem. One of the things when you move forward in new directions, you're really talking about ideas and concepts that have no precedent. True innovation is doing something that has never been done before. So that means you can't go to a manual, you can't go to paper, you can't go look at well-developed methodologies and implement it. You actually have to carve and create that pathway. So you can take all your can'ts and you can put them out there as challenges by making data access available to anyone. And then what will happen is that you can get groups of researchers coming together, finding solutions, and putting it out there in the public domain. And what I liked about the challenge structure is that you may have a winner or two, but it turns out you probably will have, from all these different solutions, a framework that emerges. And you can use that framework to set your next challenge, and then your next challenge, and your next challenge. And iteratively, by the time you get to the 10th, 15th challenge, you've carved that path. You've turned that can't into a can. And that's what we really see the value of bringing together all this data in all its messiness and then pushing it forward through equal access, through the Data Atlas, through the Alzheimer's Disease Data Initiative, for whatever data sharing platforms there are out there, and invite the community to create these solutions. So we're very excited about sort of this opportunity. So if we can go to the next slide. So I really wanted to end this by just sort of asking people to think about not what we already know, but think about how technology is really going to help us push forward to really develop new directions, new ideas in ways we've never even thought about and probably can't even conceive today. And what I'd like to do is invite all of you to join us in this effort. Thank you very much.
And we actually do have about 5 minutes left in your talk, Rhoda. So if anyone wants to put a question in the chat, we can go ahead and answer that live.
I mean, I could mention to you, just to let you know, we do have actually, so we've been very strategically targeting low to middle income countries. And so we have currently signed letters of commitment from cohorts from countries that include Malaysia, the Philippines, Taiwan, Singapore, India, Argentina, Brazil, and Panama. And then we've also engaged Neuronet, which has a very large reach within the European cohorts. And so they have signed on as sort of a consortia partner where they're bringing on, they've identified 15 different sites and have specifically targeted participants that meet sort of our criteria of being racially, ethnically diverse, because that's really our focus in terms of how to move forward. And so we're very excited because we're going to start with the digital phenotyping, as I described, and that's where we're really hoping that the IHCC and particularly those that I think that Patrick's going to discuss in the next presentation would be interested in coming on board and being part of this effort to really expand into many different cohorts, regardless of the resource environment, in order to bring in this digital phenotyping as an initial step for equalizing the playing field. So I hope that helps to just give some perspective. You know, the IHCC is really about obviously large scale cohorts, but we're really targeting how do we bring in these much smaller cohorts. And I think that that was sort of also what's been mentioned before is how the IHCC has an interest in really engaging sort of cohorts that are much smaller in size, but are certainly much, can help sort of with the global representation of the work you're doing. So I hope that helps.
Yes, and it looks like we do have a question from Ricardo Verdugo, which is shown now on the screen.
Oh, so a fluidic digital biomarker. So that's sort of a term I've decided to just create. And the reason I did that is because I've been very concerned about the fact that there's been a lot of what I would say limited use or limited conceptualization of a digital biomarker. So one of the things that we tend to do is we like to anchor against what we know. And usually when we think of biomarkers, we think of things like blood-based biomarkers, for instance, or in my world, imaging biomarkers, like from PET scans, et cetera. And usually when you talk about, you know, sort of biologically based biomarkers, you're talking about a particular way of measurement and a particular signal that you're looking for. It's consistent and it's reliable, and you're always looking for that as your indicator of disease or symptom or whatever. In the realm of digital, that's not going to work because, first of all, the kinds of data we're collecting are very dynamic and very heterogeneous. And as we're trying to think about where's the value of digital, it's really on sort of in the earlier stages of a disease. So in the case of Alzheimer's disease, it's an extraordinarily heterogeneous disease with multiple etiologies. That's why we have identified lots and lots of different risk factors that impart risk. So what's going to happen is that if you use the current conceptualization of a biomarker for digital, you're going to miss your mark. So I'm going to give you an example. When you have someone who might have Alzheimer's disease, when you bring them in for the initial intake, you know, it's very common to ask someone, when did you first notice symptoms? And to ask their family members as well. And they can often date it years earlier. And here's what will happen. They'll give you sort of a series of evidence of what led them to believe that they're now experiencing a persistent memory impairment. So they might say things like, well, I forgot to turn off the stove, and I get lost when I go to a familiar place, and I, you know, forget my keys, or I forget, you know, and it turns out that it's not that that person always forgets to turn off the stove, always gets lost when they're in a familiar place, always misplaces their keys. There's nothing always. And if you ask the family member, they will come up with a different compilation of symptoms that led them to believe in the memory impairment. And that's what digital is going to have to do. A digital biomarker is going to have to be dynamic. It's never going to be the same, and it's going to change on an ongoing basis. And it's really about the pattern of which it comes together in this dynamic, fluid way. And that's what's going to be the digital biomarker. So I really have tried to come up with this term as a way to conceptualize digital biomarkers very differently from how we think of current biomarkers. So I hope that helps.
Great. I guess I'm up. So I hope everyone can hear me. I'm going to be discussing some efforts that we've been leading here at SHOP around polygenic risk scores and IHCC, and taken this into a pilot project for Alzheimer's with Davos. So many of you will be aware of the background to this talk. Over the past 15 years or so, thousands of GWAS studies, covering practically all common and complex phenotypes, have now reported genetic variants that are associated with the risk of developing those diseases. Depending on the phenotype, these variants can number, from the tens to the hundreds, but they typically confer a modest risk per allele. And so polygenic risk scores have been proposed as a method to aggregate these small risks into a score, which may be more clinically actionable. But this depends on the phenotype. And so applying a PRS across a cohort that will result in a normal distribution of the scores, and it's the individuals at the extreme right-hand side of the distribution, those who are carrying the most alleles, the risk alleles, that will be considered in the high-risk group. Constructing a PRS is very heavily dependent on having a well-powered GWAS to identify both of your SNPs and their weights. Knowing population LD structures can differ. Both the risk SNPs themselves and these effect sizes can differ across populations. And so designing a well-tuned PRS that will be applicable globally is kind of the challenge. One of the reasons this is a challenge is because so many of the majority of genome-wide data sets that have been generated so far have been generated in people of European ancestry. So if we take a look at the GWAS catalog, it's a website that sort of catalogs GWAS studies over the past that have been carried out and published. Around three quarters of those are listed as having participants of European descent. Even though their European ancestry global population accounts for about 16%. So these numbers in the catalog, and that was a quick search that I did, they don't add up to 100. It's participation rather than total. These are supported by independent estimates. So there's a couple of papers here that I've listed that come up with similar numbers. And overall, what we see following the European ancestry cases is that Asians are the second most commonly represented population in GWAS studies, followed by African Americans and Hispanic Latinos. Unfortunately for us, developing PRSs, transancestry studies are the most informative to generate the scores from, so that they're globally applicable. And these, unfortunately, only account for about, less than 1% of publications. So the beginning of 2020, we started a pilot project with the IHCC, and really this was to set the stage for the mechanism, really, putting in place the figuring out how to do this logistically. And we selected 4 phenotypes. 2 were common traits, a BMI and a blood pressure, and 2 were complex diseases. We chose type 2 diabetes and asthma. They were mainly chosen because of their commonality across populations, and also because we thought that they would be relatively well represented amongst the cohorts. So apart from setting the stage, we had a scientific goal, and what we wanted to do was to look at the efficacy of a transancestry score and compare that with individual ancestry-specific scores. And this is really leveraging the power of IHCC with the global population of that's represented, allowing us to do this. So the structure that we designed was a little bit involved. We generated SNPs and weight files at SHOP for the 4 main continental ancestries. So those would be African, Asian, Hispanic, European, and the 5th was then the transancestry weight files. We then disseminated those to the participating sites, and we asked each cohort to generate scores using both the transancestry weights and the ancestry-specific weights that would most closely match their population. We then collated those output metrics and compared them. So in the pilot, we had great representation from groups across the globe. We had participants from Brazil, Norway, the US, China, and the UK. And the groups are listed in this table. And briefly, what we found was that the transancestry PRS actually outperformed the ancestry-specific PRS in all groups except except the Europeans. So leveraging the power, the greater power of the European GWASs improve the PRSs across all other groups. It did reduce the power slightly in the Europeans themselves, but given that you've developed a score that's globally applicable, it's actually it's actually not that much of a hit that's worth worrying about. Other things that we learned, so the general approach of developing the scores at one site and applying them across the consortium. Now we had some issues, and I think that we learned a lot on how to improve that, but it did work quite well for the pilot, and we actually managed to do this in a very short amount of time. And then we learned a few things that were perhaps not surprising. One is that the more data we have, both across populations and phenotypes, the better. And the other is that there is increased value in sharing more data for the purpose of analysis across the consortium itself. And so, next slide, please. So moving on to the current proposal, we're looking at a slightly different approach here, and it's a simplified approach. So we have a global population of participating sites, and the UK Biobank is a reference. So what we're proposing to do is to construct two different PRSs, a trans-ancestry score and a European score that will be generated from one of the large meta-analyses that I'll discuss soon. So the endpoint that we'll be looking at is looking at the correlation of these PRSs with measures of cognitive function from AD or dementia rating scale assessments. Next slide, please. So, as I said, one of the main things that determines the success of a PRS is what data is available. So if you take a quick look at what is available, we have some recently published very large-scale meta-analyses. For example, IGAP have published a paper recently with just over 53,000 cases, and they identified 21 genome-wide significant loci from that, 18 of which had been previously reported. So that's a paper by Kunkel et al. Then we have a paper by Janssen et al. They included 3 different independent consortia. The PGC Alzheimer's Working Group, they had the International Genomics of Alzheimer's Project, IGAP again, and the Alzheimer's Disease Sequencing Project, ADSP. They did this in phase 1, and then in phase 2, they included the UK Biobank data. So they totaled 71,000 cases and reported 29 risk loci. And then the largest paper so far that I've been able to identify is a European effort by Belenguez et al. They included the European Alzheimer's Disease Biobank data and the UK Biobank in phase 1, and then ADGC, FinGen, Charge in phase 2, for a total of just under 86,000 cases. Now, this brings the total number of loci up to 75, 33 of which were previously reported and 42 novel. The one thing that's common across all of these studies, and you can see that they actually share some of the same data, is that they're all also exclusively European ancestry participants. Luckily, there is one recent, relatively well-powered trans-ancestry genome-wide study that was published by June et al. From the ADGC. So that included European ancestry, African Americans, Japanese, and Israeli Arabs. So a fairly good representation across the globe. Now, the authors in this paper didn't actually generate a PRS or report one. What they were interested more in is using this data to look at the the risk conferred by ApoE alleles and how it varies by ancestry. And it does vary quite considerably. So compared with the Europeans, the epsilon 4 allele frequency is lower in Asians and associated with higher AD risk. And the opposite is true in African Americans, where E4 is increased about 50% higher than European ancestry, and it confers lower risk. Next slide, please. So to summarize, it seems like we actually have some good summary data available, and heritability estimates in AD are high. They range from 60 to 80% estimates. So do we have any, do we know how well PRS performs in Alzheimer's disease? And we do have some data, mainly from Europeans again. So the large European study by Belenguez did evaluate a PRS that they generated from their data and a number of population-based studies. And what they found was, so they constructed their PRS out of 83 genome-wide significant SNPs. It's not one of these, PRSs that includes many million SNPs. They didn't include ApoE. But this small instrument is actually significantly associated with both progression to all-cause dementia and Alzheimer's disease. And it's also significantly associated with MCI progressing to all-cause dementia and Alzheimer's disease. The p-values, if you're interested, have been included there. They're pretty significant. So there is less data on the transethnic scores, but luckily there is one paper, a recent paper by Sofer et al., and they compared PRS generated from two of the European large meta-analyses that we just discussed, the Janssen PGC and the Kunkel-IGAP papers, against, I guess, together with June transethnic GWAS data and a GWAS from the FinGen group. And the FinGen data largely centered around the ApoE locus. Now, their target population was an admixed Hispanic cohort with MCI from the Seoul Inca study. Well, what they found, interestingly, was that the smaller admixed paper and the FinGen study, which identified the ApoE loci, SNPs at the ApoE locus, were actually more predictive in their admixed population compared with the larger European ancestry-derived data. So on the right-hand side here are the odds ratios. You can see that the first is the FinGen set of data, and the third is the June et al. And in all but the Amerindians, these are positively associated compared with the European data. Next slide, please. So that brings us to a decision: which data do we use? So to avoid overfitting this PRS model, we have to ensure that the pilot sites that are going to participate in this study haven't contributed their data to the reference GWAS studies. And this can be an issue with ever larger meta-analyses. And in fact, it does rule out two of the European meta-analyses. Both the Belenguez and Janssen studies did include the UK Biobank data, which we're going to use as our reference cohort. So that leaves us with the Kunkel data for the European score. And for the transancestry score, the June et al. Data is the largest available transancestry GWAS. And we know, based off of the Sofer data, that the PRS developed from that should outperform European-derived scores in non-Europeans. But they just applied this to Hispanic, so it remains to be seen if this applies to African-American and Asian populations. So far, we have another great set of representation from the network. We have interest from ELSA Brazil, SAPRIN in South Africa, the Korea Biobank Project, Singapore National Precision Medicine Program, the East London Genes and Health Study. This recruits Britons of South Asian ancestry study, and the National Center for Geriatrics and Gerontology from Japan, us at CHOP. And we have potential participation from the Southeast Asian cohort study. Again, the UK Biobank is going to be used as our reference cohort. So the inclusion criteria for this AD-PRS are quite simply availability of the genotypes and AD phenotype endpoints. And while these cohorts have sort of shown some interest, we do invite all cohorts to participate. Next slide, please. So as before with the IHCC BMI pilots, what we're proposing is a federated model for this analysis. So sites will generate their PRS locally and share the performance metrics with a CHOP. And the reason we do this is because it avoids issues of data transfer, et cetera, which while solvable do make everything slower. So CHOP will then generate the SNPs and weight files for the two PRSs, the European and the ancestry scores, and we'll share those across the network. Sites will be responsible for their own imputation and APOE phasing. And we'll list with some standardized covariates that we would like the sites to generate. So the deliverables from this will include, this is from our end, a detailed experimental approach, and we'll propose QC metrics. We'll suggest reference panels to use and bioinformatic pipelines. And the aim here really is to achieve as much consistency across the cohorts as possible to reduce the experimental variability. So we'll provide an analytical framework so it's possible to collate and compare the outcomes across the cohorts, and where possible, we'll meta-analyze the data that we have in our hands. Next slide, please. So this project is ongoing. As you can see, we've already started reaching out to cohorts, and we're developing a plan. So we aim to have the PRS algorithms and the SOPs ready to share and disseminate by the end of this month. Sites will then have between 4 and 6 weeks. We'll obviously give sites as much time as possible to generate the scores and return those metrics to CHOP. From that point, we'll collate and finalize the cross-cohort analyses and deliver those back to the DAC by July 31st. And we're aiming for a publication on this pilot by the end of this year. So thank you for your attention, and happy to take any questions.
Hello, everyone. I have the pleasure today to give you an update on behalf of the working group on data infrastructure and interoperability. So just to remind you, our working group works on combining cohorts to build a IHCC cohort atlas, and there are a number of challenges involved. As you imagine, there's very limited documentation to find out what data cohorts are available around the world for sharing. In addition, there's also a lack of standardization and harmonization of the data, which can come in many forms. There could be questionnaires, genomic data, images, is really a wide variety of data. And also because of jurisdictions in terms of data protection and regulation, sometimes the data is not allowed to be moved around from one country to another. In addition, there are lack of standards for phenotypes and health outcomes. And then, of course, there is a difference in terms of cultural differences in terms of risk tolerance and privacy. So in the Cohort Atlas projects, we want to bring together several axes of the cohort data in order to make discovery. So for example, disease status, data use, sample collection parameters, genotypes and phenotypes around the population cohorts. So we try to gather a diverse set of over 100 cohort data dictionaries, and we're aiming to survey and collate these data dictionaries for all the IHCC cohorts, harmonize the metadata in a semantic way, and also make available an online atlas in order to support cross-cohort queries in order to facilitate discovery. So in order to do this, we had to build a framework to get all this data harmonized and loaded into a cohort atlas. We always do not want to reinvent the wheel. So in our case, we tried to make use of standards and tools and reuse components from other projects in order to satisfy these three-step process. First of all is to build the data models to represent both the data access and the metadata of the cohorts. We also need tools and processes in order to implement. And lastly, we want to apply these tools and processes and data models to all the clinical cohorts. So I'm going to show you some of the tools and processes that we're using from existing projects in order to build out this IHCC cohort atlas. So the first step is about data models, and there's actually two components in it. There's about data use and also about the actual data model for the metadata. So first of all, data use. We want to codify the consent that were given to the data set so we know what the data can be used for. So for example, a data may be consented for general research use, or maybe it can only be used for specific disease-specific research. All this can be codified using the GA4GH standard. So here an example where the European Genome Phenome Archive has put together the codes and along with the terms of use for those data sets, and this can easily be searched and matched to a data user request. If you want to see more examples, you can go to any of these links and also get the source code as well. And the second part is actually building the data model for the metadata. So Gecko is an output from the Seneca project. The project has tried to harmonize the metadata from multiple cohorts and find the commonly used attributes to build out the core data model that can be extended out to different projects or different disease types. And then this can be used for the next step. The next step is to build out the tools and processes in order to get the cohort data into a registry and mapping. Next, please. So for the registry, in order to build out these human-readable cohort dictionaries, we use existing tools, primarily from the EBI team. They have built out these toolkits for semantics. And so here's an example where the cohort dictionaries are loaded into a registry. And version control is already built in, and also there's automatic detection when updates made. And there are also lookup services. So this is really a great reuse of existing tools at EBI so that actually the next steps can reuse additional tools from the toolkit. So here's an example of a reuse of a tool called OXO. This allows the data from the cohorts to be mapped to the Gecko data model. And you can see from the mapping that inference can be made. So for example, if cohort A, some terms from cohort A can be mapped to cohort B, and cohort B can map to cohort C, then maybe you can make an inference between A and C. And these kind of mappings are publicly available through an API, so you can go to the site and take a look at the mappings. Next, please. As you can all imagine, mapping the data dictionaries to the data models is actually very time-consuming, and it is often the bottleneck. So there's a strong need to automate these mapping and put the tool in the hands of the cohort owners so they can do the initial round of the work. And if they have trouble, then the harmonization team can come in and help. So the EBI team has built out this tool so that cohorts owners can submit Excel spreadsheet to the tool. Those spreadsheets are then processed. Initial mapping is done. Then the result can be viewed by the data owners. There could be warnings, or they could be areas that could not be mapped. And data owners can make changes accordingly and go through the revalidation again. And this is an iterative process to get the mapping. And eventually, when they're happy with the results, they can now push send, and the data mapping will go into the cohort registry in the back end. So again, this is leveraging existing software the EBI team calls Zuma, and this is all Dockerized and deployed in their existing infrastructure. And then the next step, of course, is apply these processes and techniques to clinical cohorts in IHCC. So next, please. So we have done this with initial set of cohorts. There are 5 cohorts. They were focused on diversity. So these cohorts are from Africa, Genomics England, Iran, Korea, and South Africa. So just to give you an sort of an end-to-end view of the process that we, in order to harmonize a data dictionary from a cohort and get it added into the atlas. Let's start from the right-hand side, where we use the Gecko data model for the metadata. We take in the data dictionary and massage it into the right shape using the tools that I've just described. And this gets processed into the data registry. And then we also add in some of the cohort admin data. This is provided to this IHCC Secretariat. And then both these data are combined into format that can be exported and consumed by the Atlas, which is the web interface that you can search. So this is a process that we have now finalized and can do it in a fairly quick way. Next slide, please. So this is the IHCC Cohort Atlas that you can see. The URL is at the bottom right. This is already in production. Again, for this web search interface, we also reuse software we have already built for other projects. So specifically, the software suite is called Overture. This has been used for other projects such as the International Cancer Genome Consortium. So this search interface is readily available using the modules, the search API as well that we can reuse. For our initial set, like I said, we have 5 cohorts deployed, and we're in the process of adding 7 more cohorts. I do hope this user interface is intuitive, so you don't need a walkthrough, but just very quickly, just mention that usually on the left, there are a set of cohort metadata and data dictionary attributes, so you can do your search. For example, you may be interested in finding cohorts that collect sleep habit data, so you can find that. The cohorts that meet your search criteria will show up in the middle as a table. The graphics at the top will also update based on your search criteria to give you a very high-level summary. And then right now we have the external links to the cohort sites if you're interested in finding more information. So you may actually remember this atlas from last year's summit. So last year in May, when we first presented the first version, it took us about 3 months to get the initial 5 cohorts and this web interface built. But now we have spent the time to harden the infrastructure and harden the pipeline. We can now very easily get a new cohort into the Atlas. So as an example, we have reused this pipeline early this year on the Davos Alzheimer's Collaborative Cohort Atlas. Rhoda gave you an overview of Davos earlier, and this is the Atlas we've built for Davos. The initial cohort has about 12 cohorts, and there are a lot more data variables specific to Alzheimer's diseases. We were able to build this within a month, from ingesting the dictionaries to making them searchable in the cohort, on the web. So if you're interested in this, you can go to these links to take a look. This atlas is not officially in production yet, but hopefully this will be released sometime soon. So lastly, I want to make a shout out. We have really spent the time to make this data ingestion and data harmonization process easier and speedier. We just want to encourage all the cohort stakeholders to get your data ready and reach out and add your cohorts to the Atlas. And so at the top here, this is the email you can reach us if you're interested to get a head start. Any one of you will be happy to give you a general overview, maybe a demo, and then help you along the way to get your data into the Atlas. Lastly, I want to thank the team here. Thomas and Philippe are the co-leads for the working group. The OICR team is responsible for the web interface for the search. The EBI team are really the lead in developing many of the tools for data harmonization and ingestion. So thank you all for your attention.
Good morning. So my name is Håkon Larsson. I'm the director of the Center for Applied Genomics here at Children's Hospital, and I'm a co-chair of the working group for the scientific strategy within the IHCC. And I'm going to review with you this morning the scientific strategy and cohort enhancement group work for the last year. And this is a collaborative between myself, Adam Butterworth, and Gad Rennan. The outline for this overview is to, we spent actually a fair amount of time last year on the Davos Alzheimer Collaborative in terms of coming up with the feasibility of that and the foundation phase. So I'm going to review that with you briefly. We worked with the Atlas for the IHCC for cohort mapping. We worked out a scientific strategy for funding 6 projects that were actually presented to you earlier this morning. And we have worked on revisiting the previous proposals, many of which were actually quite compelling, and have been looking into taking them into new directions. So the Davos Collaborative, this was really a focus to build a global, pan-global sort of diverse cohort with minority populations worldwide, and to sort of show that contribution of data from diversity minority populations would or could contribute to discovery within Alzheimer's disease, because as Rhoda and others have indicated before, the vast majority of previous work has actually been conducted on European ancestry populations. So there was extensive foundation work that took place last year. This was co-chaired by myself and Dr. Elias Zerrouni, former NIH director. And we pulled together over 70 experts in the field of AD, essentially across all disciplines. And we looked into phenotypes, omics, data from sequencing, genotyping, and through RNA-seq, epigenetic factors, etc., clinical biomarkers, digital biomarkers, and other informative sort of biomarkers to guide and inform on the program. And this culminated into essentially sort of a white paper that was presented to the DAC management this past, or presented by the DAC management, Dr. George Rattenberg and Drew Hofsfeld, to the World Economic Forum, which founded sort of the DAC structure back in 2020, and this was the annual meeting in 21, and a pilot project to conduct a foundation phase was subsequently launched thereafter. So the principal focus was on research diversity cohorts globally, pilot funded, as I said, and sort of, really to establish that this group could work together, and we could work with the IHCC cohort that had expressed interest in working with us based on surveys that we have conducted to move this program forward. And then DAC has sort of launched a 5-year plan for the future upon completion on the foundation phase. And if you go to the next slide, this is sort of just the schematic for the foundation phase, basically to look into what is the added value of diversity populations with respect to contribution to understanding the omic landscape in Alzheimer's disease compared to standardized European ancestry sort of data sets such as the UK Biobank. And we had these 3 pillars that we focused on, and the first one was to create sort of, a summary of data from the existing data that existed within these cohorts, and we sort of anticipated we would do this for a population of maybe 12, 15,000 individuals or greater. And then pillar 2 was to sort of gap fill with information, new data that was missing within the cohorts that would be funded through the DAC program to bring together sort of unified, comprehensive data. And the third phase was sort of future biobanking, which we actually put quite a bit of effort into and came up with a white paper for the structure and management of a centralized biorepository for future research purposes. So this sort of initial phase, as Patrick just presented, was decided to focus in on the polygenic risk score to identify individuals within these diversity and minority cohorts who had been phenotyped to some extent for cognition with either rating scales for dementia or other measures, and then pull in additional data that would sort of, enrich that phenotype perhaps further with risk factors, known clinical risk factors for Alzheimer's disease. And then there is a whole list here of information and data that we surveyed, and the numbers that we are presenting here, which are actually in the millions, where data is available across the IHCC cohorts, not for everybody, but for a subset of patients that cumulatively comes up in a very, very large numbers. And so the goal, again, create a uniform minimum data set that would be useful, valuable to identify individuals that would be at greatest risk. We would subsequently invest in for sequencing data, for imaging data, for clinical and digital biomarker data. And if you go to the next slide, there were a number of cohorts that expressed initial interest that came through with the surveys. We did actually 3 surveys around data that existed and data that would be possible to collect, and certainly with focus of interest on the right sort of age range of the patient populations. And as you can tell, the outcome of that from this initial sort of assessment was quite robust, with a couple of million people sort of available, and over 500,000 people available with phenotypic information in the age range of 50 to 60 years of age, which we felt was the prime sort of interval to assess the phenotype for risk, and use polygenic risk to subsequently take out the most extreme and follow them over time to look at their cognitive decline in those individuals who would be at greatest risk for that. This is the pillar 2, sort of just to sort of emphasize the gap filling with additional data that we anticipated that we would not need much for the European cohort because those already exist. But for South America, Middle East, East Asia, Southeast Asia, and Africa, we would need to generate data for exome sequencing, imaging, phenotyping, and biomarker data for both phenotyping and cognitive assessment. And this is sort of just an estimate for the first sort of a round of numbers that we would focus in on in these different territories in the world to come up with a comprehensive, coherent data set that we could utilize and do at least a head-to-head comparison with the value added from those data sets compared to the European data sets. The polygenic risk score, Patrick already reviewed this with you, which is already work underway, and the algorithms is just about to be disseminated now to the cohort sites for execution. There certainly will be opportunities to bring in many additional cohorts, but this initial pilot phase, we're trying to sort of generate an early hit to demonstrate that we can have the cohorts work together. And we can deliver on that within a short period of time. So there is a meeting in August that was looking into sort of presenting upon those first feasibility data to demonstrate that this system at the IHCC level works, and is worthy future funding for this particular purpose. And then, of course, any sort of gaps in the data sets, as I said, we can propose what would be needed to complete a comprehensive data set for that purpose. So if you go to the next slide, Patrick sort of really emphasized or went through the polygenic risk, but it's really focused in on ultimately to identify the individuals who are at the highest polygenic risk site, but we should also keep in mind there are going to be individuals who are at the very lowest polygenic risk site. These are potentially individuals who are protective for development of Alzheimer's, so they can be actually equally important to generate data on to better understand protective factors across omics, imaging, and clinical and digital biomarker data. And the polygenic risk would be validated. We would generate a trans-ethnic score, as Patrick mentioned, and meta-analyze the data across the cohort, and then focus in on the individuals who would be permitted to have a callback that had permitted recontact based on IRB approvals and would be willing to participate in future research. And given the numbers, even though the attrition is obviously going to be significant, we believe we will have a sufficient number of individuals to generate meaningful data in that setting. And again, this ultimate goal is to set the feasibility for this program and identify individuals we can invest in with additional data. Next slide. I believe this is now item number 2 in terms of the cohort mapping, and we heard from the Atlas group already this morning, but the scientific strategy group did some pre-work for this program to enable that in terms of coming up with the directory of data to be uploaded and imported into the Atlas. And again, numerous cohorts have already signed up and completed the mapping, so those data registry data are in the Atlas already and can be reviewed, as we were shown earlier this morning, or in the previous talk. And more and more cohorts are coming on board, and these are all in the works, and others are being added on for this effort. Next slide. So the pilot projects that got funded, this was actually a significant scientific, really strategic effort to come up with a competition essentially for cohort leaders to submit proposals, and there were numerous proposals that came in. This was scored by an independent group unrelated to any of the projects submitted, and 6 programs were funded. And we heard, I think, from most of them this morning. First one listed here on exploring the genetically determined BMI in infancy, and carry that through the lifespan is an interesting one for the development of cancer and other conditions later in life. The Metabolomic program, as we heard from the Nightingale biomarker measures, is just about to be released now. The Opioid project is making good progress, and so is the global COVID program, as well as the global biobank collection in the for the longitudinal biorepository data that John Chambers presented. And the coronavirus psychiatric susceptibility is from the host factor is led by Jordan Smoller no, sorry, by Michelle Ramsey and the pandemic mental impact from COVID by Jordan Smoller and Harvard. We did revisit some of the, we felt were some of the more interesting project ideas that had come up from the meeting in Iceland now a couple of years ago, and sort of look to the risk factors in low-income countries and addressing the diversity gap, certainly important during these particular times we've lived now. And we believe the structure and sort of the way the IHCC cohorts are set up, I mean, we are in a prime sort of situation of executing on this, and with funding that could be provided to genotype, ethnically sort of specific populations by SNP arrays that could be selectively generated for population where we don't have such data today, and or by a low sort of depth whole genome sequencing data that we would be able to subsequently utilize for the purpose of understanding population structure and variations that are population specific. Loss of function variants, similarly, I mean, that was certainly an area that we felt was very robust in such a large scale diversity cohorts as IHCC presents, and the ability to tag and identify and collect individuals with loss of function variants and subsequently phenotype them more specifically with some outside funding from the agency would be certainly highly relevant and important to understand sort of the biological functions of the loss of function genes, and obviously PCSK9 and some other projects are prime examples where this type of a research has led to really a very meaningful therapeutic development. For future directions, there will be more discussion sort of about later on, both today and tomorrow, in terms of the scientific strategy that we have taken within IHCC and how we see that being implemented. But this is sort of the focus areas. Really, the sky is the limit here, if you think about it, because I mean, with such large number of cohorts of the scale and data that is available, doing cross cohort analysis, phenotype harmonization for GWAS, CNV, exome genome analysis, and looking into epigenetic proteomics and other biomarker factors would obviously allow for a very powerful integrative analysis of data, which would be very unique and not be something that any individual group could do, but IHCC could do, and obviously pursue grant funding and collaborations maybe with pharmaceutical developers and others, sort of for the solving targets that may be population specific or more globally applicable. That time will tell for that. So I think this is my last slide. If you go to the next one. So thank you for the audience.
Thank you all. I was provided with a few minutes here today, really just to review the outcomes of the discussions that we had last year when we went through each of the policy documents that were developed by the Policy and Biodata Sharing Working Group more in depth, and then we had a period of work over the summer and through the fall on comments. So I'm going to just walk us through quickly, and if I can just have the next slide, I'll get the disclaimers out of the way, just to say that I am here representing my own views and not those of my organization. Again, my name, which I don't think I said it was up on the screen, Laura Rodriguez. I am currently working at the Patient-Centered Outcomes Research Institute in Washington, D.C., but am participating as a long-term member of the IHCC. And so I'm very excited to be here thinking about these issues with you this morning. Next slide. We will, as I said, give a quick review of the policy documents as a foundation for not just the conversations that we heard this morning, but really, I think, moving into the discussions that we will have over the next day and the day after that as well, for thinking about the future of IHCC projects and the guideposts that we will use for our work as we do that, and probably will not spend much time talking about other updates given the time. So next slide. Okay, so as I mentioned, there have been updates since we dove in depth into the 3 documents that the Policy and Biodata Sharing Working Group put together for last year's discussion at the summit. And we did receive several very helpful comments from IHCC members following the summit, and had some exchanges with them as well on those. And so just wanted to give a shout out to them again for all of the time that they provided and the thought that they did to help make sure our policies were really sound and represented all of the different perspectives within IHCC. These policies were provisionally approved by the current steering committee and will be presented to, as we'll talk about tomorrow, the newly constituted scientific steering committee once that group is fully up and running. Next slide. So as you might recall, as I mentioned, we had 3 different policy documents, all dealing with core elements to guide IHCC's work. And we also took an approach using different policy tools, if you will, for how we approached the different topics based on what the topic was. So the first of our documents was a full-fledged policy. So these bring with it, if you will, sort of the strongest tool that we have yet deployed in terms of expectations and structure for how the policy will guide IHCC activities. This is, of course, focused around publication, which is obviously fundamental to what IHCC will do and to our core value to make sure that the findings and results from IHCC projects are disseminated broadly. The policy walks through the different types of publications, consortium publications, project publications, etc., discusses, again, the priority of publishing in a timely way and in open access formats, also provides guidance and, again, some structure with regard to publication working groups, authorship contributions, highlighting, as we'll talk about based on comments, the desire to make sure that we are being inclusive and recognizing junior investigators as well, recognizing all of the cohorts and the participants that participate within the cohorts in our acknowledgement statements, and again, also providing an example draft template for developing in advance a publication and authorship plan, which should come forward with any proposal for an IHCC scientific project. Quickly, again, changes that were made based on the discussions that we had to do with clarifying language in terms of what the different policy types were. We're explicit, as I mentioned, about encouraging the inclusion and recognition for junior investigators, both in terms of the roles that they played and their ability to be lead authors on publications coming out of IHCC projects. Added the expectation to explicitly recognize research participants in every IHCC publication, as well as the cohorts and the lead investigators for those cohorts. And then finally also made an explicit allowance in the timeline that is laid out for the publication and authorship plan for organizational review of publications, since that was important in some of our IHCC member organizations. And so we wanted to make sure people were planning for that. We'll move over to the second document. And in this case, the tool that was used was to develop guidance for how IHCC projects and members would collaborate with industry. Again, the value that we wanted to express through this particular policy document was our inclusion and expectation to promote IHCC work by collaborating with all sectors within the research enterprise that could bring value and advance the scientific opportunities and or resources available to IHCC projects, as well as building capacities for IHCC cohorts where that would make sense. Within the guidance document, again, written as guidance to recognize the desire for consistency and congruence in all of our different collaborations, but also allow for flexibility within them. There are details with regard, again, to the different types of collaborations that might be possible among the different parties, whether they are IHCC member cohorts or non-participating IHCC organizations who have an interest in a particular project, etc. Principles upon which the collaborations should be built are, again, around bringing value for scientific opportunity, recognition and opportunity to participate in an inclusive way from all cohorts, and again, potentially building capacity where that would be appropriate for our cohorts as well. This guidance is structured then according to a series of points to consider, which walk through the different values established within our membership agreement for IHCC as an organization as a whole, with some specific points to keep in mind. And then a checklist is provided as well as a resource for members, but the actual collaborative agreements would, of course, be between the parties participating in the collaboration and not directly with the IHCC, which again supports the rationale for this particular policy strategy to be done as a guidance with a checklist to assist members, but not to dictate the specific format or content of every collaborative agreement. We'll just again review the changes made, again, very much focused on being explicit with regard to the different kinds of interests and value that could be brought to IHCC through these partnerships, clarifying edits made with regard to when the guidance applies and when it might not apply, such as when IHCC members are participating in their own independent activities with the private sector, et cetera. Modified some of the language within the principles, and again, just clarified also aspects of what was expected with regard to transparency, which is, of course, another fundamental value within IHCC for all of our activities, to recognize the fact that there could be proprietary agreements set up among the parties in these collaborative agreements. And so what was expected within this guidance was a summary to be publicly posted on the IHCC website for the substance of the agreement, but not the specifics. This moves us then to our final document, and perhaps not our most fundamental, but certainly equally fundamental activity in terms of promoting data sharing for the value that can be brought for the entire research enterprise as well as IHCC itself by having a value around open science and making sure that our data are available not only for the IHCC work, but also for secondary work as appropriate going forth. So here again, as much as data sharing is fundamental to everything that IHCC will do and how we come together in our collaborations, it is also incredibly complex as we all come from different parts of the world. And so we have different individual cohort policies, local and national legal frameworks within which we must work, as well as our varying cultures and the standards and expectations within those cultures for how data sharing should be conducted and overseen. And so in this case, the approach used by the Policy and Biodata Sharing Working Group was to provide principles, core principles, both foundational principles, which would be basic expectations for IHC activities, as well as aspirational principles, wherein this would point to our larger, longer-term goals around data sharing and data access. The expectations are that the principles work together. So as you review these now or in the future, understand that they're all incredibly interdependent, and that was fundamental to being able to think through these issues. Likewise, again, something else that the Policy and Biodata Sharing Working Group, but as we've heard about from others today, that IHCC as whole is looking to do is not to reinvent the wheel when there's already a good wheel out there, but to build upon and expand the good work of others. And so again, our data sharing principles rely upon the basic Global Alliance for Genomics and Health framework as an ethical foundation for sharing data, and then add these principles on top of that for the specific IHC context. The foundational principles, of course, speak to the primacy of consent and local policies, the legal frameworks that must be in place, ensuring that there's robust data management and data security always present. And then the aspirational principles will speak more to optimization of data access processes and data sharing capacities for secondary research purposes, et cetera. Many of the things that the Data Atlas is also beginning to work on by building a platform through which we can hopefully affect some of these data sharing principles in the future. Changes were really in this regard around clarifying what the principles were addressing and how they were intended to be used. Also noting very clearly that these principles were meant to be a living document and that they would be updated over time, since we all know that data sharing and the cultural context for data sharing is changing. We've seen it change a lot just in the last year in terms of the public conversation as well as the scientific and research conversations with this regard. The other important thing to note is that these principles are a launching pad, if you will, for developing project-specific policies, again, based on the nature and context of the individual cohorts that are participating in any particular project. I do just want to acknowledge that we've had several additional members join our working group, including, I'm very happy to announce, Nikki Tiffin, who has come on as our new co-chair for the Policy and Biodata Sharing Working Group. And so I'm very excited to have her. And I also want to give a huge shout out and thank you to Gunn-Peggy Knudsen from Norway, who has been my co-chair for the past 3 years and had to step away as COVID responsibilities and changes in her other work just simply made it impossible for her to give this the time that she wanted to. But she was a huge help and really foundational to all of the work that has been done to date. And I welcome Nikki for what we will do going forward. So with that, I will stop and turn it back to Adam.
Thanks very much, Laura. Sorry the time was a bit up against you, but I'm glad we were able to hear you in the end. And thank you to all the wonderful speakers today. It's been hugely inspiring, such an enormous amount of work. By my count, I think we have projects now including more than 40 cohorts from more than 20 countries, including many low and middle income countries, spanning 6 continents. So enormous breadth of work, as we've heard as well. And I think the way that such an enormous consortium could rapidly pivot to address several aspects of COVID research is incredible. I think we also heard as well about some of the challenges as well. And I really want to think we should take forward Rhoda's mantra that there are many apparent can'ts in the way, and how do we turn those into challenges that we can address? And I think in the last series of talks there, we've heard excellent summaries of the 3 main IHCC working groups and the progress they've made in the last year, particularly to address some of the challenges around data sharing, data interoperability and harmonization. And I've seen lots of comments in the comments section about how we might take some of these forward, both engaging more cohorts in scientific projects, expanding the amount of cohorts in the Atlas, and how we can engage the IHCC user community more. As well as thanking the speakers, we should also again just thank the people behind the scenes who've been making the show run smoothly, particularly Ida and Hannah and Ludi. So thank you for a day that has worked technically. We've had great slides, great talks, and it's all been recorded. Do get in touch with those folk if you have any challenges. Finally, just to say tomorrow, good timing. We start again at 11 Universal Time. Look forward to a keynote from Mary De Silva from the Wellcome Trust. And as was alluded to today, there's been a lot of work that's gone on in the past several months thinking about the strategic vision, the next 5 years of IHCC. And we look forward to introducing that to you tomorrow and also engaging you. So please join the breakout sessions. I think you've all been asked to nominate a breakout session you'd like to join. We need your help. We want you to help inform the action plans. So how do we implement the strategic vision in respect to the scientific working group, the data working group, the policy working group, and the new learning and education working group. So we need your help. And we look forward to engaging with you in a more interactive way during those breakout sessions. And then in session 2, we'll hear more about efforts to involve a greater diversity of cohorts. How can we fill some of the gaps and overcome some of the barriers, and taking that challenge attitude into the next phase of IHCC. So with that, I'll just thank again all the speakers, thank all the participants for your attendance. I hope you found the day stimulating and challenging, and look forward to seeing you all again tomorrow and interacting with more of you. And do feel free to stay around in the Socio platform and use that to connect with speakers or other participants to get the most out of the meeting. Thank you very much, and see you all tomorrow.